Hello and welcome to the Late Lab. It's lovely to have you here tonight. Before we begin, I'd love to know where in the world you're listening from. A quiet bedroom with the lights dimmed? A comfortable sofa after a long day? A late-night train ride somewhere in the dark between cities? Wherever you happen to be, it's always interesting to see where these calm explorations are reaching. And tonight's topic might feel particularly fitting. Because if you're watching this in the evening, or perhaps already feeling a little tired, then something rather interesting may already be happening inside your brain. But before we drift too far into tonight's subject, please take a moment to like the video and subscribe to the channel if calm journeys through science are something you enjoy. And to those already subscribed, thank you. Your support genuinely helps this sleepy little laboratory keep going. Now, sleep is something every human being experiences, yet it remains surprisingly mysterious. Every night the mind slowly loosens its grip on the outside world, muscles relax, thoughts begin to wander, and eventually the brain performs something rather unusual. It deliberately lets go of consciousness. For roughly a third of every human life, the brain enters this strange state. From the outside, it looks quiet and still, but inside the skull, the brain is often remarkably busy. Neurons shift their rhythms, memories may be reorganized, and subtle biological processes begin working quietly through the night. And despite decades of research, scientists are still trying to understand exactly how this happens. So tonight, we're going to explore that question together. Not just why sleep exists, but how sleep actually works. Every night, something rather unusual happens to nearly every complex animal on Earth. Awareness fades, muscles relax, and behavior slows until the body settles into a state of stillness. For hours at a time, the brain allows the organism to become quiet, unresponsive, and in many cases surprisingly vulnerable. From a purely evolutionary perspective, this behavior looks strange. A creature that cannot easily react to danger would seem to be at a disadvantage. Yet across mammals, birds, reptiles, and even some very simple animals, something recognizably similar to sleep appears again and again. This raises a question that has puzzled scientists for centuries. If survival depends on constant awareness, why would evolution preserve a state where awareness disappears for roughly a third of life? At first glance, sleep seems simple. Close the eyes, lie still, and rest. But the more closely researchers have examined the phenomenon, the more complicated it has become. Sleep is not merely rest. It is a carefully organized biological state, produced by intricate coordination inside the brain. Long before modern neuroscience existed, thinkers were already trying to explain what sleep might be. Aristotle suggested that sleep occurred when warmth from digestion drifted upward toward the head, producing heaviness in the senses. Later physicians in the 17th and 18th centuries believed sleep was linked to circulation changes in the brain. These early ideas were speculative, but they shared an important assumption, that sleep must have a physical cause rather than a mystical one. By the 19th century, neurologists began examining sleep more seriously as a biological process. Researchers observed that brain injuries could dramatically alter sleep patterns, suggesting that specific regions of the brain might regulate it. Gradually, the idea emerged that sleep might be controlled by the nervous system itself rather than by digestion, fatigue, or simple inactivity. Even so, for a long time, scientists believed the brain more or less shut down during sleep. The prevailing assumption was that the sleeping brain simply rested, conserving energy while consciousness paused for the night. That assumption turned out to be wrong. In the early 20th century, a young physiologist named Nathaniel Kleitman began investigating sleep with unusual persistence. Working at the University of Chicago, Kleitman believed that sleep should be studied with the same careful measurements used in other areas of physiology. Rather than relying on speculation, he wanted to observe what the brain and body were actually doing during the night. This idea may sound obvious today, but at the time it was remarkably ambitious. Sleep research required scientists to monitor people for long periods while they were unconscious, something that was technically difficult in the 1920s and 1930s. Kleitman and his students often slept in laboratories themselves while conducting experiments, turning their own nightly rest into data. Over years of careful observation, Kleitman helped transform sleep from a philosophical curiosity into a measurable biological phenomenon. His laboratory examined breathing patterns, body temperature changes, and behavioral rhythms across the day and night. The emerging picture suggested that sleep was not random. It followed structured patterns, controlled by internal biological timing. One of Kleitman's students, Eugene Aserinsky, would later make one of the most surprising discoveries in sleep science. While studying the eye movements of sleeping volunteers, Aserinsky noticed something unexpected. At certain times during the night, the eyes moved rapidly beneath the eyelids, darting back and forth as if tracking something invisible. These episodes became known as rapid eye movement sleep, a phase of sleep in which the brain becomes highly active. In fact, during these periods, the brain's electrical activity can resemble the patterns seen during wakefulness. This discovery forced scientists to reconsider a fundamental assumption. If the brain was truly resting, why would it sometimes become more active during sleep? The answer revealed something profound. Sleep was not simply the brain switching off. It was the brain entering a different mode of operation. Modern neuroscience now shows that sleep involves carefully coordinated changes across many parts of the brain. Networks of neurons shift their electrical rhythms. Chemical signals adjust the balance between alertness and rest. Entire brain regions change how strongly they communicate with one another. In other words, sleep is constructed. The brain actively produces it through a complex interplay of biological systems. Signals rise and fall throughout the day, gradually increasing the pressure to sleep. Internal clocks track the passing hours and influence when the body feels ready for rest. When these forces align, the brain begins guiding the body into sleep through a series of controlled transitions. From the outside, this process looks quiet and uneventful. Yet inside the skull, billions of neurons are reorganizing their activity. Certain brain regions reduce their responsiveness to external stimuli. Others begin producing slow electrical rhythms that sweep across large areas of the cortex. Chemical messengers alter the behavior of entire neural circuits. Muscles relax, breathing slows, and consciousness gently dissolves. The result is the state we recognize as sleep. And once the brain enters this state, it does not remain static. Instead, the night unfolds as a sequence of shifting brain conditions, each with its own patterns of activity and biological functions. Some phases emphasize deep restorative processes. Others are associated with vivid dreaming and intense neural communication. Taken together, these stages form a complex biological cycle that repeats throughout the night. What makes this especially intriguing is that sleep appears across a vast range of species. Elephants sleep only a few hours each day, while bats may sleep for most of the daylight hours. Dolphins can allow one half of their brain to sleep while the other half remains awake, enabling them to surface for air. Even fruit flies display periods of inactivity that closely resemble sleep. Such widespread behavior strongly suggests that sleep performs fundamental biological roles. If sleep were merely a luxury or an optional form of rest, evolution would likely have removed it long ago. Instead, it has persisted across hundreds of millions of years of evolutionary history. This persistence hints at something deeper. Sleep may not simply be beneficial, it may be necessary. The difficulty is that no single explanation fully captures why sleep exists. Some researchers believe sleep restores brain chemistry that becomes unbalanced during wakefulness. Others argue that sleep reorganizes neural connections, helping the brain refine what it has learned during the day. Still others propose that sleep allows the brain to clear away metabolic waste that accumulates during normal activity. Each of these ideas contains evidence, yet none alone explains the entire phenomenon. What becomes increasingly clear is that sleep is not a simple biological event with one purpose. It is a complex system serving multiple functions at once. While consciousness fades, the brain continues working quietly in ways that waking awareness rarely notices. The central mystery, then, is not merely why sleep exists. The deeper question is how the brain orchestrates this transformation so reliably, night after night. How does the brain know when to begin shutting down wakefulness? What mechanisms coordinate the shifting stages of sleep? And why does the biology of sleep appear so deeply embedded in the architecture of the brain itself? Answering those questions requires looking more closely at the hidden systems that control when sleep begins, because before the brain can enter sleep, it must first build the pressure that makes sleep unavoidable. If sleep were controlled by a single switch somewhere in the brain, the mystery would be much easier to solve. One signal would turn it on, another would turn it off. Simple, but biology rarely works that neatly. Instead, the brain manages sleep through a delicate balance between two powerful forces that slowly build and interact across the day and night. One measures how long the body has been awake, the other keeps track of time itself. Together, these two systems quietly negotiate when sleep will begin. Sleep scientists often describe this framework as the two-process model of sleep regulation. The model was proposed by the Swiss researcher Alexander Borbély during the 1980s, and it remains one of the most influential ideas in modern sleep science. Borbély suggested that sleep emerges from the interaction of two biological processes. The first is sleep pressure, sometimes called process S. The second is circadian timing, sometimes called process C. Sleep pressure behaves exactly as the name suggests. The longer a brain stays awake, the stronger the pressure to sleep becomes. This pressure builds slowly and steadily throughout the day. Imagine the brain as a machine that accumulates tiny molecular traces of wakefulness. Each hour of thinking, moving, reacting, and perceiving leaves behind microscopic signs of activity inside the nervous system. Among the most important of these signals is a molecule called adenosine. Adenosine forms naturally as neurons consume energy. Every time a brain cell burns fuel to send electrical signals, small amounts of adenosine are produced as a byproduct. Over the course of waking hours, adenosine gradually accumulates in brain tissue, and as its concentration rises, something interesting begins to happen. Neurons become less eager to fire. Communication between brain cells becomes slightly slower. Mental processing feels heavier. Attention drifts more easily. Reaction times stretch a little longer than usual. The sensation we recognize as sleepiness begins to appear. In simple terms, adenosine acts like a biochemical record of how long the brain has been active. The longer the waking period continues, the more adenosine builds up. Eventually, the brain begins encouraging rest. Experiments in neuroscience laboratories have confirmed this pattern in remarkable detail. Researchers studying brain activity in animals have observed adenosine levels rising steadily during wakefulness and falling again during sleep. The implication is straightforward. Sleep helps clear the chemical traces of waking activity. Remove the adenosine, and the brain can reset its readiness to think clearly again. But the story becomes even more interesting when an everyday substance enters the picture. Caffeine. Caffeine does not remove adenosine from the brain. Instead, it interferes with the receptors that normally detect it. These receptors sit on the surface of neurons, waiting to bind with adenosine molecules. When caffeine occupies those receptors, adenosine can no longer deliver its signal effectively. The chemical message of fatigue is partially blocked. As a result, the brain temporarily feels more alert, even though the underlying sleep pressure continues to build. Eventually, the caffeine fades, and the accumulated adenosine makes itself known. The familiar afternoon crash begins to make sense. Yet sleep pressure alone cannot explain why people often grow sleepy at predictable times of day. Many individuals feel tired late in the evening, even after a quiet, restful afternoon. Others feel strangely alert late at night despite long hours of wakefulness. Clearly, something else is influencing the timing of sleep. This brings us to the second force in Borbély's model: circadian timing. Circadian rhythms are biological cycles that repeat roughly every 24 hours. These rhythms influence a wide range of physiological processes including hormone release, body temperature, metabolism, and alertness. Deep within the brain sits a tiny cluster of neurons known as the suprachiasmatic nucleus. This structure acts as the body's primary biological clock, synchronizing daily rhythms with the external cycle of daylight and darkness. Throughout the day, this internal clock sends signals that influence wakefulness and sleepiness. During daylight hours, the circadian system promotes alertness. Hormones and neural signals help the brain maintain focus and energy, even as sleep pressure slowly builds in the background. Later in the evening, the circadian signals begin shifting in the opposite direction. The body gradually prepares for sleep. Body temperature begins to drop, hormone levels change. Neural circuits that promote alertness slowly reduce their activity. When the rising sleep pressure from adenosine finally meets the circadian signal that encourages rest, sleep begins to feel almost inevitable. The brain has reached a biological tipping point. Researchers have explored these interacting forces in carefully controlled laboratory experiments. Sleep scientists such as Robert McCarley and Alan Hobson studied patterns of brain activity during sleep and wakefulness using electroencephalography, a method for recording electrical signals from the brain. Their work revealed that sleep does not appear randomly. Instead, it emerges when biological pressures and timing systems converge. Sleep deprivation studies make this even clearer. When people remain awake for unusually long periods, the pressure to sleep becomes overwhelming. Attention falters. Reaction times slow dramatically. In extreme cases, brief episodes of microsleep can occur, during which the brain slips into sleep for a few seconds, even while the person appears awake. The brain simply refuses to remain conscious indefinitely. And when sleep finally arrives after prolonged wakefulness, the body often compensates by entering deeper sleep stages more quickly. This phenomenon, sometimes called sleep rebound, demonstrates how strongly the brain prioritizes restoring balance. The lesson from these experiments is striking. Sleep is not triggered by a single signal. It emerges from the interaction of multiple regulatory systems working together. Sleep pressure tracks how long the brain has been active. Circadian timing tracks the passage of day and night. Chemical signals and neural circuits coordinate the transition between wakefulness and sleep. The result is a carefully balanced biological system that guides the brain through its daily rhythm of alertness and rest. And once that balance tips towards sleep, an entirely different set of processes begins to unfold inside the brain. There is a quiet clock inside the brain. It does not tick audibly, and it has no moving hands, yet it keeps time with remarkable precision. Day after day, year after year, this biological clock organizes when the body feels alert, when it grows tired, when hormones rise or fall, and even when body temperature subtly shifts. Long before alarm clocks, calendars, or artificial lighting living, organisms were already following internal rhythms that repeated roughly every 24 hours. Scientists call this system the circadian rhythm, from the Latin phrase meaning about a day. Circadian rhythms influence far more than sleep. They help regulate metabolism, control hormone release, shape patterns of alertness and fatigue, and even influence how the immune system behaves across the day and night. The human body, in many ways, is built around this repeating cycle. Cells throughout the body adjust their behavior depending on where they believe the organism is within the daily rotation of light and darkness. At the center of this timing system lies a tiny structure deep within the brain. It is called the suprachiasmatic nucleus. This small cluster of neurons sits within the hypothalamus, just above where the optic nerves cross. Despite its modest size, containing only around 20,000 neurons, the suprachiasmatic nucleus acts as the master biological clock for the entire body. It coordinates daily rhythms across organs, tissues, and neural systems, ensuring that countless biological processes remain synchronized. The discovery of this structure transformed how scientists understood circadian biology. During the 1970s, researchers studying brain lesions in animals noticed something unusual. When the suprachiasmatic nucleus was damaged, the animals lost their regular daily patterns. Sleep and wakefulness became irregular. Activity levels drifted unpredictably through the day and night. The internal sense of time had effectively disappeared. When the structure remained intact, those rhythms returned. The conclusion became clear. This tiny group of neurons was acting as the body's central timekeeper. Yet the suprachiasmatic nucleus does not operate in isolation. It must remain aligned with the external world, particularly the daily cycle of sunlight and darkness. If the internal clock drifted too far from the environment, the body's rhythms would slowly lose synchrony with the actual day. Fortunately, the brain has a mechanism for keeping its clock properly adjusted. Light. Specialized cells within the retina of the eye detect changes in brightness and transmit that information directly to the suprachiasmatic nucleus. These cells are known as intrinsically photosensitive retinal ganglion cells, and they contain a light-sensitive pigment called melanopsin. Unlike the rods and cones responsible for vision, these retinal ganglion cells are not primarily concerned with forming images. Instead, they measure the overall brightness of the environment. When daylight reaches the eyes, these cells send signals along a pathway called the retinohypothalamic tract, delivering information about the external light cycle to the brain's clock. This daily signal allows the suprachiasmatic nucleus to adjust itself continuously, ensuring that internal biological time remains aligned with the rising and setting of the sun. The precision of this system is remarkable. Even in constant darkness, human circadian rhythms tend to continue running for the period very close to 24 hours. This persistence shows that the rhythm is generated internally, rather than simply imposed by the environment. Yet exposure to regular daylight gently nudges the clock each day, keeping it synchronized with the real world. Behind this steady rhythm lies an intricate molecular mechanism. Inside the neurons of the suprachiasmatic nucleus, genes and proteins interact in feedback loops that create oscillating patterns of activity. Certain genes are switched on, producing proteins that gradually accumulate inside the cell. Once these proteins reach a particular level, they suppress the very genes that created them. As the proteins break down over time, the genes become active again, beginning the cycle once more. This self-regulating loop forms a biological oscillator. Two scientists who played a major role in uncovering these genetic mechanisms were Joseph Takahashi and Michael Young. Through decades of research, they helped identify several key genes that drive circadian rhythms, including genes commonly known as clock, period, and cryptochrome. These genes act as components of the molecular clock, producing proteins that rise and fall in repeating cycles across roughly 24 hours. Their work, along with contributions from researchers at institutions such as Rockefeller University and the Howard Hughes Medical Institute, revealed that circadian timing is embedded directly within the genetic machinery of cells. In other words, the body's sense of time is written into its biology. This discovery helped explain why circadian rhythms appear throughout the body, not just in the brain. Liver cells, immune cells, and even skin cells possess their own local clocks, each following similar molecular rhythms. The suprachiasmatic nucleus acts as a conductor, coordinating these many cellular clocks so that the entire organism stays in harmony. As the day progresses, the circadian system subtly changes the body's internal environment. Hormone levels shift. Body temperature gradually rises during daylight hours, supporting alertness and cognitive performance. Later in the evening, temperature begins to fall slightly, helping prepare the body for rest. Hormones such as melatonin are released from the pineal gland when darkness increases, signaling that nighttime has arrived. These signals do not force sleep to occur immediately. Instead, they prepare the brain for it. Meanwhile, another force has been quietly building throughout the day. As neurons remain active, adenosine continues accumulating, increasing the biochemical pressure for sleep. By evening, this pressure may already be substantial. When circadian signals begin encouraging rest at the same time that sleep pressure has reached a high level, the brain approaches a kind of biological agreement. The internal clock suggests that night has arrived. The accumulated chemistry of wakefulness suggests that the brain has been active long enough. Together, these forces gently push the nervous system towards sleep. The timing of this interaction explains why people often feel most alert during the daytime even if they woke up early, yet become noticeably sleepier later in the evening, even after relatively quiet activities. The circadian system helps maintain alertness for much of the day, despite growing sleep pressure, then gradually allows that pressure to take effect once darkness arrives. In this way, the brain's internal clock does not merely track time, it shapes the entire rhythm of waking life. And when its signals finally align with the accumulated need for sleep, the transition from wakefulness towards sleep begins to unfold. If sleep were controlled by a single master switch, the brain's nightly transition into unconsciousness would be simple. One signal would silence wakefulness, another would restore it, and the system would operate much like a light being turned on and off. But the brain does not work like a lamp. Instead, sleep emerges from a complex negotiation between groups of neurons that promote wakefulness and others that promote rest. These networks communicate through chemical messengers called neurotransmitters, and their interactions determine whether the brain remains alert or gradually slips into sleep. One of the most important of these messengers is gamma-aminobutyric acid. Gamma-aminobutyric acid, often abbreviated as GABA when scientists discuss it informally, is the brain's primary inhibitory neurotransmitter. Its role is to quiet neural activity. When neurons release gamma-aminobutyric acid, the receiving cells become less likely to fire electrical signals. The effect is similar to gently lowering the volume across a network of communicating brain cells. In regions that promote sleep, this inhibitory chemical plays a crucial role. Clusters of neurons within the hypothalamus and nearby brain regions release gamma-aminobutyric acid as the body prepares for sleep. These neurons target parts of the brain responsible for maintaining alertness, gradually reducing their activity. As the inhibitory signals spread, wake-promoting circuits begin to quiet. The sensation is familiar, thoughts slow, concentration drifts. The mind becomes less responsive to external stimulation. Eventually, the balance tips. Yet these sleep-promoting neurons do not operate alone. The brain also contains powerful systems dedicated to maintaining wakefulness. These networks release their own neurotransmitters, encouraging alertness and cognitive engagement throughout the day. Among these chemicals are dopamine, serotonin, and acetylcholine. Dopamine is often associated with motivation and reward, but it also plays a role in maintaining wakefulness. Dopamine-releasing neurons in parts of the midbrain help sustain attention and goal-directed behavior. When dopamine signaling remains strong, the brain tends to stay alert and responsive. Serotonin contributes to mood regulation and emotional balance, yet it also participates in sleep and wake cycles. Neurons in the brainstem release serotonin in patterns that influence both wakefulness and the early phases of sleep. Acetylcholine, another key neurotransmitter, becomes particularly active during states of attention and learning. High levels of acetylcholine in certain brain regions help maintain the vivid mental activity associated with waking consciousness. These wake-promoting systems form a powerful network. They encourage the brain to remain engaged with the world. However, there is another system that plays a stabilizing role in this delicate balance between sleep and wakefulness. Its discovery dramatically changed how scientists understood sleep disorders. In the late 1990s, two research groups working independently made a remarkable discovery. One group was led by Emmanuel Mignot at Stanford University. The other was led by Luis de Licea, also studying neural signaling pathways related to alertness. Both teams identified a small population of neurons that produced a previously unknown neuropeptide. This molecule became known as hypocretin, sometimes also called orexin. Hypocretin neurons reside within the hypothalamus, and although their numbers are relatively small, their influence is widespread. These cells send projections to many regions of the brain involved in maintaining wakefulness. By releasing hypocretin, they help stabilize alertness and prevent sudden transitions into sleep. The importance of this system became clear when researchers studied a neurological disorder known as narcolepsy. Individuals with narcolepsy experience sudden and uncontrollable episodes of sleep, sometimes occurring in the middle of ordinary activities. In severe cases, laughter or strong emotions can trigger abrupt loss of muscle control, a condition known as cataplexy. When scientists examined the brains of people with narcolepsy, they found something striking. The hypocretin-producing neurons were largely missing. Without these stabilizing signals, the boundaries between sleep and wakefulness became unstable. The brain could slip suddenly from one state into another, producing the unpredictable symptoms seen in narcolepsy. This discovery demonstrated that sleep regulation is not simply a matter of turning sleep on or off. Instead, the brain maintains a dynamic balance between systems that promote alertness and systems that promote rest. One of the clearest explanations of this balance was proposed by the neuroscientist Clifford Sapir at Harvard Medical School. Sapir introduced a concept often described as the flip-flop switch model of sleep regulation. In this model, sleep-promoting neurons and wake-promoting neurons inhibit one another in a mutually opposing relationship. When the wake-promoting network is active, it suppresses the sleep-promoting network. When the sleep-promoting network becomes dominant, it suppresses the wake-promoting system. This arrangement produces a stable outcome. The brain tends to remain firmly in one state or the other. Rather than drifting slowly between wakefulness and sleep, the system shifts relatively quickly once the balance changes. This helps explain why people often feel alert one moment and noticeably sleepy the next, as if an internal threshold has been crossed. The flip-flop switch also reduces the likelihood of unstable intermediate states where both systems might remain partially active. In engineering terms, it creates a cleaner transition between two modes of operation. Yet even this system does not operate in isolation. Signals from the circadian clock influence when sleep-promoting neurons become more active. Meanwhile, biochemical sleep pressure generated by adenosine continues building during wakefulness. These external pressures slowly push the neural balance towards sleep. When the combined forces become strong enough, the inhibitory signals from sleep-promoting neurons begin suppressing the wake-promoting networks more effectively. Gradually, the brain relinquishes its hold on wakefulness. What emerges from this intricate system is not a simple command structure, but a distributed network of interacting circuits. Sleep is not ordered by a single control center, issuing instructions to the rest of the brain. Instead, it arises from cooperation and competition between many neural systems working simultaneously. Some neurons encourage alertness, others encourage rest, and as their chemical signals shift throughout the day, the brain moves gently from one state to the other. In this sense, sleep is less like a switch being flipped and more like a balance slowly tipping. When the scales finally settle in favor of sleep, an entirely different pattern of brain activity begins to take shape. Eventually, the balance inside the brain begins to shift. The chemical signals that promote wakefulness grow quieter. Sleep-promoting neurons strengthen their influence. Circadian rhythms tilt gently toward your rest, while the slow accumulation of adenosine continues urging the brain to pause its activity. The result is not an instant shutdown. Instead, the brain begins a gradual descent. For most people, this transition feels familiar yet strangely difficult to observe while it's happening. Awareness softens, thoughts drift, and attention becomes less firmly anchored to the outside world. Sounds grow slightly distant. The edges of perception blur just a little. The mind is still awake, but something subtle has started to change. Understanding this transition became possible only after scientists learned how to measure the electrical activity of the brain. The key breakthrough came from a German psychiatrist named Hans Berger in the early 20th century. Berger was fascinated by the idea that mental activity might produce detectable electrical signals. In 1924, after years of experimentation, he succeeded in recording the brain's electrical rhythms from the human scalp using a technique that would later be known as electroencephalography. Electroencephalography, often shortened in laboratories to the initials EEG, measures tiny fluctuations in electrical voltage produced by groups of neurons communicating with one another. Although the signals are extremely small, sensitive instruments can detect them through electrodes placed gently on the scalp. For the first time, scientists could watch the living brain at work. What Berger discovered was remarkable. Even when a person sits quietly with eyes closed, the brain produces rhythmic patterns of electrical activity. These repeating patterns appear as waves on an electroencephalogram recording, rising and falling in smooth oscillations. One of the most common patterns during relaxed wakefulness is known as the alpha rhythm. Alpha waves typically occur at a frequency of roughly 8 to 12 cycles per second. They are most prominent when a person is calm, awake, and not actively processing complex sensory information. Closing the eyes often strengthens the alpha rhythm, suggesting that the brain is entering a more relaxed mode of operation. As the transition towards sleep begins, something interesting happens to these rhythms. The orderly pattern of alpha waves gradually fades. In its place, slower and less regular electrical patterns begin to emerge. These new rhythms are known as theta waves. Theta activity occurs at a lower frequency, typically around 4 to 8 cycles per second, and it signals that the brain is entering the earliest stage of sleep. The change is subtle but measurable. Neurons that once fired in relatively organized patterns during wakefulness begin coordinating their activity differently. Communication between brain regions becomes less tightly synchronized with external sensory input and more influenced by internal rhythms. In simple terms, the brain begins turning its attention inward. During this period, the senses gradually lose their dominance over consciousness. Sounds from the environment may still be detected, but they carry less significance. The brain becomes less inclined to respond. A faint noise might be noticed but quickly ignored. Visual input also fades as the eyes close, and the visual cortex reduces its engagement with the outside world. This process is sometimes described as sensory disengagement. The brain is not completely cut off from its surroundings, but it is becoming increasingly selective about which signals it allows into awareness. Strong or unexpected stimuli can still provoke a response, yet ordinary background sounds begin slipping past unnoticed. Meanwhile, the mind itself undergoes curious changes. Thoughts become less structured. Instead of deliberate reasoning or focused attention, the brain begins generating fragments of imagery, memory, and abstract impressions. These drifting mental experiences often resemble the early stages of dreaming. A person might briefly imagine a scene, hear a fragment of conversation, or feel as though they are starting to move through a space that does not quite exist. Then the thought dissolves. Another appears in its place. Psychologists sometimes describe this stage as hypnagogia, a transitional mental state between wakefulness and sleep. Hypnagogic experiences can include fleeting images, distorted perceptions of time, or the sensation of falling just as sleep approaches. Many people have experienced the sudden jolt that occasionally occurs during this transition. Just as the body begins to relax completely, a brief muscle contraction may occur, sometimes accompanied by the sensation of slipping or falling. These events are known as hypnic jerks, and although their exact cause remains debated, they appear to be related to the nervous system adjusting its control over muscle activity during sleep onset. For most individuals, this entire process unfolds quietly within a few minutes. Yet from the perspective of brain activity, a great deal has changed. The orderly rhythms of wakefulness have softened. Neural communication is shifting toward patterns that favor slower, more synchronized activity. sensory input is losing its influence over conscious awareness. The brain is preparing to move deeper into sleep. Researchers studying electroencephalography recordings noticed that this early phase marked only the beginning of a much more complex sequence. As sleep progresses, the brain continues transitioning through several distinct stages, each characterized by its own patterns of electrical activity and physiological behavior. In this earliest stage, however, the brain has only taken the first step. Wakefulness has begun to loosen its hold, but sleep has not yet reached its deeper forms. The mind remains capable of returning to full awareness with relative ease. However, sudden noise or a gentle nudge can quickly restore alertness. The threshold between wakefulness and sleep is still thin, but once the brain crosses that threshold more completely, the electrical rhythms of sleep become stronger and more organized. Slow waves begin to dominate the cortex, and the structured architecture of the sleeping brain begins to reveal itself. Once the brain moves beyond the fragile threshold between wakefulness and sleep, the night does not unfold as a single continuous state of unconsciousness. Instead, sleep reveals a surprisingly organized structure. The brain cycles through a repeating pattern of distinct states, each with its own electrical rhythms, physiological changes, and functional roles. Rather than simply remaining asleep, the brain moves through a carefully choreographed sequence of activity that repeats several times across the night. This pattern was not immediately obvious when scientists first began studying sleep. Early researchers assumed that once sleep began, the brain simply remained in a stable resting condition until morning. Electroencephalography recordings revealed something very different. When scientists examined the electrical activity of sleeping volunteers, they noticed that brain rhythms were constantly changing. Periods of deep, slow activity would gradually give way to faster, more irregular patterns, and then return again to slower rhythms. Sleep was not static, it was cyclical. One of the most important discoveries came from the observation of rapid eye movement sleep. Researchers noticed that certain periods of sleep were marked by quick, darting movements of the eyes beneath closed eyelids. These episodes appeared alongside distinctive patterns of brain activity that looked surprisingly similar to wakefulness. The brain, it seemed, was entering alternating states. Scientists began dividing sleep into two broad categories. The first became known as non-rapid eye movement sleep, often abbreviated as NREM in scientific writing. The second was rapid eye movement sleep, or REM sleep. Each of these states possesses its own characteristic brain rhythms and physiological behavior. Non-rapid eye movement sleep includes several progressively deeper stages. During these stages, the brain's electrical activity becomes slower and more synchronized. Large groups of neurons begin firing together in rhythmic patterns, producing the slow waves visible on electroencephalography recordings. Rapid eye movement sleep, by contrast, shows faster and more complex electrical activity. In many ways, the brain appears almost as active as it is during wakefulness. Yet despite this activity, the body remains largely immobilized due to inhibitory signals that temporarily suppress muscle movement. This contrast between brain activity and physical stillness is one of the many peculiar features of sleep. Over time, sleep researchers developed a standardized system for classifying these stages. Today, many sleep laboratories follow guidelines established by the American Academy of Sleep Medicine, which organizes sleep into distinct stages based on characteristic patterns observed in brain recordings. These stages include lighter phases of non-rapid eye movement sleep, deeper slow wave sleep, and rapid eye movement sleep. Each stage contributes something different to the overall architecture of the night. What makes this system especially interesting is how predictably it repeats. A typical night of sleep is organized into cycles that last roughly 90 minutes. During each cycle, the brain gradually moves from lighter sleep into deeper, slow-wave sleep, before eventually transitioning into rapid eye movement sleep. After this phase concludes, the cycle begins again. Over the course of a full night, most people experience several of these cycles. Yet the structure of these cycles changes as the night progresses. Early in the night, deep slow-wave sleep tends to dominate. The brain produces large, powerful oscillations known as delta waves, reflecting highly synchronized neural activity across the cortex. These slow waves are associated with the most restorative phases of sleep, when the body's physiological systems perform much of their repair and recovery work. Later in the night, rapid eye movement sleep becomes more prominent. Dreaming often occurs during these later cycles, when brain activity becomes faster and more varied. The cortex engages in complex patterns of communication, and the sleeping mind can generate vivid internal experiences. This gradual shift from deep restorative sleep toward more dream-rich phases reflects a carefully regulated biological rhythm. The brain appears to allocate different functions to different periods of the night. Modern imaging technologies have helped scientists examine these transitions in greater detail. Functional brain imaging studies conducted by researchers at institutions such as the National Institutes of Health have shown that different brain regions change their activity dramatically across sleep stages. During deep, slow-wave sleep, areas of the cortex become highly synchronized, producing the slow oscillations visible in the electroencephalography recordings. Meanwhile, certain regions associated with external sensory processing become less active. During rapid eye movement sleep, the situation changes again. The visual cortex and emotional processing centers become more active, while areas involved in rational decision-making, such as parts of the prefrontal cortex, may show reduced activity. This unusual combination of activation and suppression may help explain the strange and often illogical narratives that occur during dreams. What becomes clear from these observations is that sleep behaves less like a uniform state, and more like a sequence of carefully coordinated brain modes. Each phase represents a different pattern of neural communication. Each stage carries its own biological significance. Together, these stages create the architecture of the night. This architecture ensures that sleep is not merely passive rest. Instead, it allows the brain to move through a series of internal environments, each supporting different processes. Some stages emphasize restoration of neural networks. Others appear to support memory consolidation or emotional processing. The night, in other words, is structured. The brain follows a repeating pattern that balances multiple biological needs across the hours of darkness. Understanding this structure also explains why interrupted sleep can feel so disorienting. If the brain is repeatedly forced out of its cycles before completing them, the carefully organized sequence of sleep stages becomes disrupted. The body may lose access to certain phases that normally occur later in the night. When this happens, the following day often feels foggy or unbalanced. Sleep is not simply about duration. It is also about progression through the architecture that the brain has evolved to follow. Across the night, the brain quietly repeats its cycles, adjusting the balance between deep, slow rhythms and more active phases of sleep. These cycles form one of the most reliable patterns in human biology, unfolding night after night with remarkable consistency. And within these rhythms, the deepest and most powerful brain waves of sleep begin to emerge. As the night deepens, the brain begins producing some of the most distinctive electrical patterns found anywhere in human physiology. The rhythms become slower, stronger, more synchronized. Large groups of neurons across the cerebral cortex begin firing together in sweeping waves of activity that roll slowly across the brain surface. On an electroencephalography recording, these waves appear as broad, powerful oscillations rising and falling in steady rhythms. Scientists call this pattern slow-wave sleep. It represents one of the deepest stages of sleep the brain can enter, and it is often associated with the most restorative phases of the night. During this period, consciousness becomes extremely difficult to recover. Loud sounds or physical movement may still awaken a person, but the threshold for arousal is much higher than during lighter sleep. In simple terms, the brain has settled into a profoundly stable resting state. The electrical rhythms of slow-wave sleep are dominated by what researchers call delta waves. These oscillations occur at very low frequencies, typically around 1 to 4 cycles per second, reflecting large populations of neurons switching between active and silent phases together. When billions of neurons across the cortex begin coordinating their activity in this way, the result is a powerful, synchronized rhythm. For a brain that normally processes enormous amounts of information every moment of the day, this change is dramatic. During wakefulness, neural activity tends to be fast, irregular, and highly complex. Individual networks constantly communicate with one another as the brain interprets sensory information, forms memories, and guides behavior. During slow-wave sleep, the situation changes completely. Neurons enter repeating cycles of activity and silence. For brief moments, entire groups of brain cells stop firing almost entirely. These quiet intervals are sometimes called down states. They are followed by bursts of activity known as up states, when neurons briefly resume communication before falling silent again. The entire cortex begins breathing in a slow electrical rhythm. These synchronized oscillations are not random. They appear to play an important role in maintaining the health and efficiency of neural circuits. One influential explanation for their purpose was proposed by the neuroscientists Giulio Tononi and Chiara Cirelli. Their work introduced an idea known as the synaptic homeostasis hypothesis. The theory begins with a simple observation about waking life. Throughout the day, the brain is constantly learning. Every experience, every new piece of information, and every interaction with the environment can strengthen connections between neurons. When two neurons repeatedly activate together, the synapse connecting them often becomes slightly stronger. This process is known as synaptic potentiation. Over hours of waking activity, millions of these tiny adjustments accumulate across the brain. Neural circuits become more responsive as they encode memories, refine skills, and integrate new experiences. While this plasticity is essential for learning, it creates a potential problem. If synapses continued strengthening indefinitely, neural networks could become overloaded. Energy consumption would rise. Signal processing might become inefficient. Eventually, the system could lose the ability to distinguish important information from background noise. The brain, in other words, needs a way to reset. According to the synaptic homeostasis hypothesis, slow-wave sleep provides exactly this opportunity. During deep sleep, widespread neural activity becomes synchronized and simplified. This environment allows synapses across the brain to undergo a subtle process of downscaling. Connections that grew stronger during the day are gently reduced in strength. Not erased, just recalibrated. Important connections remain relatively stronger than others, preserving valuable memories and learned skills. Meanwhile, weaker or less useful synaptic changes may fade slightly, helping the brain maintain an efficient balance. In this way, sleep acts as a form of neural housekeeping. It keeps the brain's communication networks from becoming saturated. Evidence supporting this idea has emerged from several experimental approaches. Studies measuring synaptic proteins in laboratory animals show that markers of synaptic strength tend to increase during wakefulness and decrease after periods of sleep. Imaging experiments also suggest that the overall density of synaptic connections can change subtly depending on how much sleep an organism receives. These findings suggest that sleep helps regulate the enormous complexity of the brain's circuitry. Another striking feature of slow-wave sleep involves energy use. Although the brain never truly stops working, its metabolic demands shift noticeably during deep sleep. Measurements of glucose consumption and blood flow indicate that many regions of the brain reduce their activity compared with waking levels. The cortex enters a quieter mode of operation. Neurons fire less frequently overall, and communication between distant brain regions becomes more rhythmic and less chaotic. This metabolic shift may allow cells to redirect energy towards cellular repair processes and the maintenance of neural structures. The brain, which normally consumes roughly 1/5 of the body's energy, despite representing only a small fraction of its mass, finally gets a chance to operate more efficiently. Sleep deprivation. Experiments reveal just how important these slow-wave rhythms are. When people are prevented from sleeping for extended periods, the pressure for deep sleep becomes extraordinarily strong. Once sleep finally occurs, the brain tends to enter slow-wave sleep more quickly and produce stronger delta waves than usual. This phenomenon is often described as slow-wave rebound. The brain appears to compensate for lost sleep by intensifying the deepest phases of the night. The longer a person remains awake, the greater the need for these synchronized slow oscillations. Laboratory studies have confirmed this pattern repeatedly. Participants who stay awake for long periods show dramatic increases in slow-wave activity once they are allowed to sleep again. The brain seems determined to recover the processes that normally occur during deep sleep. This rebound effect offers a powerful clue about the function of slow-wave rhythms. They are not optional. They appear to be biologically necessary. Even more intriguing is the way these slow waves travel across the brain. High-resolution recordings show that the waves do not occur everywhere simultaneously. Instead, they often begin in one region of the cortex and sweep gradually across the brain like ripples moving across water. Some waves originate in frontal regions associated with decision-making and planning. Others begin in sensory areas related to touch or vision. As they propagate, large networks of neurons fall into synchronized patterns of activity and silence. It is a coordinated electrical choreography unfolding across billions of cells. And although the sleeper may appear completely still, the brain is engaged in one of the most structured patterns of activity it ever produces. These rhythms also appear to interact with memory systems. During deep sleep, regions such as the hippocampus, which plays a crucial role in forming new memories, occasionally produce bursts of activity known as sharp wave ripples. These brief events often occur in coordination with the slow oscillations of the cortex. Some neuroscientists believe these interactions allow the brain to gradually transfer memories from temporary storage in the hippocampus to more stable networks within the cortex. If that idea is correct, slow-wave sleep may help reorganize the experiences of the day into lasting knowledge. The brain, while deeply asleep, may be quietly sorting through what it learned while awake. These possibilities hint at a remarkable truth. Deep sleep is not merely a state of rest. It is an active biological process in which the brain reorganizes itself, regulates its connections, conserves energy, and prepares for the cognitive demands of the next day. The slow waves rolling across the cortex are not simply signs of inactivity. They are part of the brain's nightly maintenance cycle. And within this quiet electrical landscape, another process begins to take shape as the night continues. The sleeping brain starts to replay pieces of its waking life. Deep slow-wave sleep is not the final destination of the night. After the brain spends time immersed in those powerful synchronized rhythms, something surprising begins to happen. The electrical activity of the cortex starts to change again. Slow waves gradually fade. Neural signals become faster and less uniform. Brain regions that had been quiet begin communicating more energetically. The sleeper remains unconscious, but the brain is becoming active again. One of the most important discoveries in sleep science emerged when researchers noticed a curious physical sign accompanying this shift. Beneath closed eyelids, the eyes of sleeping volunteers began moving rapidly from side to side, darting in quick bursts as if watching something invisible. This strange phenomenon was first carefully documented in the early 1950s by Eugene Aserinsky, a graduate student working under the physiologist Nathaniel Kleitman at the University of Chicago. Aserinsky was studying sleep patterns in children when he observed these unusual eye movements during certain periods of sleep. At first, the results seemed puzzling. Why would the eyes move so actively when the sleeper was clearly unconscious? When Aserinsky and Kleitman examined brain recordings during these episodes, they found something even more remarkable. The electrical activity of the brain during these periods looked strikingly similar to patterns seen during wakefulness. Instead of the large synchronized slow waves associated with deep sleep, the electroencephalography recordings displayed faster, more irregular rhythms. The brain appeared almost awake, yet the sleeper did not awaken. These observations led to the identification of a distinct phase of sleep, now known as rapid eye movement sleep. During this stage, the brain enters a highly dynamic state, in which neural activity becomes complex and varied. Many regions of the cortex increase their activity, and patterns of communication between brain areas become more elaborate. In some ways, the brain during rapid eye movement sleep resembles the waking brain more than the deeply sleeping one. Yet there is one crucial difference: the body cannot move. During rapid eye movement sleep, powerful inhibitory signals from the brainstem suppress the activity of motor neurons that normally control skeletal muscles. This temporary paralysis, known as REM atonia, prevents the body from physically acting out the experiences unfolding within the mind. The mechanism behind this paralysis involves specialized circuits within the brainstem, particularly regions of the pons and medulla. These areas send inhibitory signals down the spinal cord, effectively reducing the ability of muscles to contract. The result is a peculiar combination of states. The brain is active. The mind may be experiencing vivid internal events, but the body remains almost completely still. This biological arrangement appears to be essential for safety. Without this temporary muscle inhibition, sleepers might physically enact their dreams, potentially injuring themselves or others. In rare neurological conditions where these inhibitory circuits fail, individuals can indeed move during dreams, a disorder known as rapid eye movement sleep behavior disorder. For most people, however, the system works flawlessly. The brain is free to wander while the body stays quiet. Within this strange neurological environment, many of the experiences commonly associated with dreaming begin to emerge. Brain imaging studies have shown that several key regions become particularly active during rapid eye movement sleep. The visual cortex, responsible for processing images during waking life, shows heightened activity. This may help explain the vivid imagery often reported in dreams, where landscapes, faces, and scenes can appear with striking clarity. At the same time, emotional centers of the brain become strongly engaged. One structure that shows notable activation is the amygdala, a region deeply involved in processing emotional significance. When the amygdala becomes active, experiences often acquire emotional intensity, whether excitement, fear, curiosity, or wonder. This heightened emotional activity may explain why dreams frequently contain powerful feelings, even when their narratives seem illogical or surreal. Meanwhile, parts of the prefrontal cortex responsible for critical reasoning and logical evaluation often show reduced activity during rapid eye movement sleep. With the brain's internal editor temporarily quieted, unusual combinations of ideas and images can unfold without the normal constraints of waking rationality. A person might find themselves walking across an ocean, speaking with someone long gone, or suddenly flying through the sky. And within the dream, these events can feel perfectly reasonable. Beyond imagery and emotion, rapid eye movement sleep appears to influence memory processing in subtle ways. Studies suggest that this phase of sleep may help the brain integrate emotional experiences from the day, reducing their intensity while preserving their informational value. Experiments conducted by researchers studying emotional learning have shown that individuals who obtain sufficient rapid eye movement sleep often process emotional memories differently than those deprived of it. The emotional charge of certain experiences may soften after a night of sleep, allowing the brain to retain the memory without carrying the same intensity of feeling. Some scientists believe that this process helps maintain psychological balance. The brain may use dreaming as an opportunity to revisit emotionally significant events within a safe internal environment. Another intriguing possibility involves creativity. Because the dreaming brain allows unusual associations between ideas, it may occasionally produce novel combinations that would not easily emerge during ordinary waking thought. Several historical anecdotes describe scientific insights or artistic ideas appearing during dreams, though such stories are difficult to verify systematically. Still, the relaxed logical constraints of rapid eye movement sleep create conditions where imaginative connections can flourish. The mind is exploring its own landscape. Despite decades of research, however, one fundamental question remains unresolved. Why does the brain dream at all? Rapid eye movement sleep clearly involves distinctive neural activity, emotional processing, and complex imagery. Yet the exact purpose of these experiences remains a subject of ongoing scientific debate. Some researchers believe dreaming reflects the brain interpreting internally generated signals. Others propose that dreams help simulate challenges or rehearse emotional situations. Still others suggest that dreaming may simply be a side effect of deeper biological processes occurring during sleep. The dreaming brain continues its quiet work through the night, weaving images, emotions, and fragments of memory into the strange narratives that so often fade by morning. And as scientists look more closely at this phenomenon, new theories continue to emerge about why these vivid inner worlds appear at all. Rapid eye movement sleep reveals a curious fact about the sleeping brain. While the body lies still and the outside world fades into quiet darkness, the mind often becomes surprisingly active. Images unfold, memories resurface. Conversations occur with people who are not present. Entire landscapes appear and disappear in moments. This strange theatre of experience is what most people simply call dreaming. For centuries, dreams were treated as messages, omens, or reflections of hidden psychological meaning. Ancient cultures often believed dreams carried divine guidance. Later, psychoanalysts such as Sigmund Freud suggested dreams revealed unconscious desires or symbolic conflicts buried within the mind. Modern neuroscience, however, approaches dreams from a different angle. Instead of searching for hidden symbolism, researchers ask a simpler and more measurable question. What is the brain actually doing while dreams occur? One influential attempt to answer this question emerged during the 1970s through the work of Alan Hobson and Robert McCarley at Harvard Medical School. They proposed what became known as the activation synthesis theory of dreaming. According to this idea, dreaming begins not with meaning but with activity. During rapid eye movement sleep, circuits in the brainstem generate bursts of electrical signals that spread upward into the cerebral cortex. These signals activate sensory regions of the brain, including areas responsible for vision, emotion, and memory. The cortex then attempts to interpret these internally generated signals. Because the brain is built to construct coherent experiences from incoming information, it begins weaving these signals into a narrative. The result is a dream. In this view, dreams are not carefully designed stories, but rather the brain's attempt to make sense of random patterns of neural activation. The mind becomes a storyteller, responding to signals that originate deep within the brainstem. A striking feature of this theory is that meaning comes afterward. The brain first generates activity, and only afterward does the cortex attempt to interpret that activity as a sequence of events. In other words, the dream may be less like a planned film and more like improvisational theatre performed by the brain itself. Yet activation synthesis does not fully explain why dreams often contain recurring themes that appear across cultures and individuals. Many people report dreams involving being chased, falling, losing control of a situation, or facing threatening circumstances. These recurring patterns led some researchers to wonder whether dreams might serve a deeper biological function. One alternative proposal comes from the cognitive neuroscientist Antti Revonsuo of the University of Turku. Revonsuo introduced what he called the threat simulation theory of dreaming. According to this perspective, dreaming may act as a form of evolutionary training. The idea is that dreams simulate dangerous situations that humans might encounter in the real world. By repeatedly rehearsing these scenarios in a safe internal environment, the brain may strengthen survival responses. Imagine the sleeping mind creating virtual environments where threats appear, and the dreamer must react. A predator approaches, a dangerous fall occurs, a confrontation unfolds. In these dream simulations, the brain may practice rapid responses, emotional reactions, and escape strategies. Although the dreamer remains physically still, the brain networks involved in perception and emotion are actively engaged. Revonsuo argued that such rehearsal could have offered evolutionary advantages for early humans living in unpredictable environments. Individuals whose brains practiced threat responses during sleep might have reacted more effectively when genuine danger appeared during waking life. Whether dreams truly function as survival simulations remains debated, but the theory highlights an important possibility. Dreams may serve purposes beyond simple neural noise. Another perspective emerges from a broader framework within neuroscience known as predictive processing. This approach is associated with researchers such as Carl Friston at University College London. Predictive processing proposes that the brain constantly builds internal models of the world. Rather than passively receiving information, the brain continuously generates predictions about what it expects to perceive. Incoming sensory data then updates these predictions, allowing the brain to refine its understanding of reality. Within this framework, dreams may represent a special case of predictive modeling. During waking life, sensory input from the outside world constrains the brain's predictions. The brain's internal models must constantly adjust to match real signals arriving from the senses. During sleep, however, external input becomes greatly reduced. With fewer sensory signals arriving, the brain may begin exploring its own internal models more freely. Dreams could represent a kind of offline simulation in which the brain adjusts, tests, and refines its predictions about the world. In this interpretation, dreaming becomes part of a larger process of cognitive maintenance. The brain is rehearsing possibilities, reorganizing internal models, and experimenting with connections between memories and experiences. The result can look strange from the inside, but the underlying purpose may be surprisingly practical. Yet another line of research focuses on emotional processing. Sleep researchers studying emotional memory have observed that experiences carrying strong emotional significance are often revisited during rapid eye movement, sleep. Brain imaging studies show heightened activity in emotional regions such as the amygdala and limbic system during dreaming. Some scientists propose that dreams help regulate emotional responses. According to this emotional regulation hypothesis, the brain may revisit emotionally intense memories during sleep in order to reduce their emotional charge while preserving the informational content of the experience. The memory remains, but the emotional sting softens. Over time, this process could help prevent overwhelming emotional reactions from accumulating within the mind. Dreams may therefore serve as a form of overnight emotional housekeeping. Memories are sorted, experiences are revisited, emotional reactions are adjusted. This interpretation helps explain why emotionally charged events often appear in dreams shortly after they occur in waking life. Still, despite decades of investigation, the scientific community has not reached complete agreement about the purpose of dreaming. Some researchers continue to view dreams primarily as a byproduct of neural activity generated during rapid eye movement sleep. Others believe dreaming represents an essential cognitive function tied to emotional regulation, learning, or survival. The truth may involve elements of several different theories. The dreaming brain is complex, and different mechanisms may operate simultaneously. Neural signals from the brainstem activate the cortex. Internal models of the world become active without sensory constraints. Emotional memories resurface and evolve. Together, these processes create the vivid mental experiences remembered as dreams. One curious detail remains particularly intriguing. People who are awakened during rapid eye movement sleep often report dreams immediately. Yet even when dreams are not remembered, rapid eye movement sleep still occurs. The brain continues its internal activity, regardless of whether the dream narrative reaches conscious recall. Dreaming may, therefore, be a constant companion to this stage of sleep, even if memory of those dreams disappears by morning. The silent theatre of the mind plays on. And while the exact purpose of dreaming continues to be debated, one pattern has become increasingly clear. Sleep is not a period of inactivity. Throughout the night, the brain is actively reorganizing information, processing experiences, and adjusting internal connections. These processes lead naturally toward another crucial function of sleep: memory itself. Dreaming may create vivid inner worlds, but another crucial process unfolds quietly beneath those shifting images. While the sleeping mind drifts through dreams or rests in deeper stages of the night, the brain is busy organizing something far more practical: Memories formed during the day are being sorted, strengthened, and gradually integrated into long-term storage. What feels like rest is actually a period of intense neurological housekeeping. The idea that sleep strengthens memory has fascinated scientists for more than a century, but the precise mechanisms began to emerge only in the late 20th century, when researchers started examining the sleeping brain in greater detail. One of the most influential discoveries came from the neuroscientist Matthew Wilson at the Massachusetts Institute of Technology. Wilson and his colleagues were studying how the brain stores spatial memories in rodents. In their experiments, rats were trained to run through mazes while researchers recorded the activity of neurons in a region of the brain called the hippocampus. The hippocampus is well known for its role in forming new memories, particularly those involving places and experiences. As the rats moved through the maze, specific groups of hippocampal neurons fired in patterns that corresponded to the animal's position along the path. These patterns effectively represented a neural map of the environment. What happened later during sleep was astonishing. When the rats fell asleep, the same neurons began firing again in nearly identical sequences to those seen during the maze exploration. The brain appeared to be replaying the experience of running through the maze, almost as if the animal were mentally retracing its steps. Sometimes the replay occurred faster than the original experience. Sometimes the sequence ran in reverse, but the essential pattern remained recognizable. The hippocampus was revisiting the day's experiences during sleep. This phenomenon became known as hippocampal replay. The discovery suggested that sleep might provide the brain with an opportunity to reinforce newly formed memories by repeatedly activating the neural circuits that encode them. Each replay could strengthen the connections between neurons, gradually stabilizing the memory trace. In simple terms, the brain seemed to be practicing what it had learned. Further research expanded this idea by exploring how information moves between different brain regions during sleep. While the hippocampus plays a critical role in forming new memories, it does not appear to serve as the brain's long-term storage system. Instead, long-lasting memories are believed to reside primarily within the neocortex, the large outer layer of the brain responsible for higher-level processing. The process of transferring memories from the hippocampus to the neocortex is often called systems consolidation. During waking life, experiences are initially encoded within the hippocampus, but during sleep, particularly during deep slow-wave sleep, the hippocampus appears to communicate these memories to cortical regions, gradually embedding them into more permanent networks. This transfer is not instantaneous. It unfolds gradually across many nights of sleep as the brain repeatedly revisits and reorganizes recent experiences. Evidence for this process has been supported by numerous human studies. One prominent line of research comes from the work of the neuroscientist Jan Born at the University of Tübingen. Born and his colleagues conducted experiments examining how sleep influences learning and memory performance. In one type of study, volunteers were asked to learn lists of words or perform complex motor tasks. Some participants were allowed to sleep afterward, while others remained awake for the same amount of time. The results were strikingly consistent. Participants who slept after learning tended to recall information more accurately and perform learned tasks more effectively than those who stayed awake. Sleep appeared to strengthen memory. Even more interestingly, the improvements were particularly strong when deep, slow-wave sleep occurred during the night following learning. This finding reinforced the idea that specific stages of sleep contribute directly to memory consolidation. But the sleeping brain does not rely on a single type of neural signal to accomplish this work. Instead, memory processing during sleep involves coordinated interactions between several different patterns of brain activity. Two of the most important patterns are known as sleep spindles and sharp wave ripples. Sleep spindles are brief bursts of rhythmic brain activity that appear during lighter stages of non-rapid eye movement sleep. These oscillations originate in circuits connecting the thalamus and the cortex, regions involved in relaying sensory information and coordinating neural communication. Sharp wave ripples, on the other hand, originate within the hippocampus itself. These ripples consist of extremely fast bursts of electrical activity that often occur during deep, slow-wave sleep. Researchers believe that sharp wave ripples represent moments when the hippocampus reactivates recent experiences. What makes this system particularly fascinating is the way these patterns appear to synchronize with one another. Sharp wave ripples from the hippocampus often occur at precisely the same time as sleep spindles in the cortex. This coordination may allow the hippocampus to transmit memory information to cortical regions, while those regions are most receptive to receiving it. In essence, the brain appears to be scheduling moments of communication during sleep. The hippocampus replays information, the cortex listens. Gradually, memory traces strengthen. Over repeated cycles of sleep, the brain refines and stabilizes the knowledge gained during waking life. The practical implications of this process are profound. Students often notice that information studied before sleep becomes easier to recall the next day. Musicians practicing new pieces sometimes find their performance improving after a night's rest. Even complex problem solving can benefit from the quiet reorganization that occurs during sleep. The brain is still learning, even while consciousness fades. Yet memory consolidation is not the only form of processing taking place during sleep. The brain also appears to adjust the emotional weight attached to certain memories, shaping how experiences are felt as well as how they are remembered. Experiences that were stressful, joyful, frightening, or meaningful may be revisited in subtle ways during the night. Sleep seems to help separate the factual content of a memory from its immediate emotional intensity. This quiet recalibration helps prepare the mind for the next day. By morning, the memory remains, but the emotional tone surrounding it may have softened or shifted slightly. In this way, the sleeping brain performs a remarkable balancing act. It preserves knowledge, strengthens learning, and gently reshapes emotional experiences, all while the outside world remains distant and quiet. The night may appear still, but inside the brain, an intricate process of remembering, reorganizing and restoring continues without pause. Memories are not the only things the sleeping brain reorganizes during the night. Emotions travel through the same landscape of neural circuits as thoughts and experiences. Joy, frustration, anxiety, relief, excitement, disappointment. All of these leave traces in the brain just as memories do. And like memories, emotional reactions do not remain perfectly stable after they occur. They evolve. One of the most intriguing discoveries in modern sleep science is that sleep appears to play a powerful role in regulating emotional intensity. Experiences that feel overwhelming during the day often become easier to process after a full night of rest. The event itself may still be remembered clearly, but the emotional charge surrounding it often softens. The brain seems to be adjusting the emotional volume. Much of this adjustment appears to involve rapid eye movement sleep, the stage associated with vivid dreaming and highly active brain activity. During this stage, emotional regions of the brain become strongly engaged, while certain areas responsible for rational control temporarily reduce their activity. This unusual balance creates a neurological environment where emotional memories can be revisited without triggering the full stress response that accompanied them originally. In other words, The brain may be reprocessing emotional experiences under calmer internal conditions. A series of influential studies from the University of California, Berkeley helped illuminate this process. The neuroscientist Matthew Walker and his colleagues conducted experiments examining how sleep deprivation affects emotional responses in the brain. In one experiment, volunteers were shown emotionally intense images while researchers measured activity within the amygdala using brain imaging technology. The amygdala plays a central role in detecting emotional significance and generating rapid responses to potential threats. Participants who had slept normally showed moderate amygdala responses to the images, but participants who'd been deprived of sleep showed something very different. Their amygdala activity increased dramatically, often by more than half compared with the well-rested group. The emotional center of the brain became significantly more reactive when sleep was missing. Even more revealing was the communication between the amygdala and the prefrontal cortex. In well-rested individuals, the prefrontal cortex helps regulate emotional responses, acting as a kind of supervisory system that can calm excessive reactions. Sleep deprivation appeared to weaken this connection. Without adequate sleep, the emotional system became more reactive while the regulatory system lost some of its influence. The result was a brain that responded more intensely to emotional stimuli. This finding helps explain something many people recognize from everyday experience. After a poor night of sleep, frustrations can feel sharper. Minor problems can appear unusually overwhelming. Emotional balance becomes harder to maintain. Sleep appears to restore that balance. Earlier research by the psychologist Rosalind Cartwright explored another dimension of emotional processing during sleep. Cartwright studied individuals going through stressful life events such as divorce or major personal upheaval. She observed that the emotional content of their dreams often reflected the challenges they were facing during waking life. Dreams became a space where emotional experiences were revisited and gradually reshaped. Over time, individuals whose dreams incorporated emotional experiences often showed better psychological adjustment compared with those whose dreams remained emotionally detached. Cartwright proposed that dreaming might allow the brain to work through difficult emotions in a symbolic or narrative form. The dream world becomes a laboratory for emotional adaptation. The brain is revisiting experiences, experimenting with interpretations, and gradually adjusting the emotional meaning attached to them. Sometimes the dream content may appear strange or abstract, but the emotional themes often echo waking concerns. Even when dreams are forgotten by morning, the underlying neural processing may still be taking place. Another important piece of evidence comes from studies linking sleep disturbances to mental health conditions. Chronic sleep disruption is commonly observed in psychiatric disorders such as depression, anxiety disorders, bipolar disorder, and post-traumatic stress disorder. While the relationship is complex, research suggests that disrupted sleep may both contribute to emotional difficulties and result from them. When the normal architecture of sleep becomes fragmented, the brain may lose some of its ability to regulate emotional memories effectively. For example, individuals experiencing post-traumatic stress disorder often show altered patterns of rapid eye movement sleep. Because this stage of sleep appears to play a role in emotional processing, disruptions to it may interfere with the brain's ability to gradually reduce the emotional intensity of traumatic memories. The result can be persistent emotional distress that remains unusually vivid and difficult to integrate. Sleep, in this sense, acts as a kind of emotional stabilizer. Across successive nights, emotional memories may be revisited repeatedly. Each time, the brain processes the experience again under calmer physiological conditions. Stress hormones such as norepinephrine tend to be lower during rapid eye movement sleep, which may allow emotional memories to be reprocessed without triggering the same physiological alarm signals. Gradually, the brain separates the memory from the emotional shock that originally accompanied it. The experience becomes something that happened rather than something that is still happening. This quiet recalibration may occur across multiple nights, slowly adjusting emotional responses as memories become integrated into broader mental frameworks. A difficult conversation may feel overwhelming in the evening. After a night of sleep, it may seem manageable. After several nights, it may become simply a memory. None of this processing requires conscious effort. The brain carries out these adjustments automatically while consciousness rests. And this emotional recalibration is only one part of a broader set of restorative processes that unfold during sleep. While the mind reorganizes memories and emotions, the body itself is undergoing important forms of physical repair and maintenance. Cells are replenished, hormones are regulated, metabolic systems are adjusted. This sleeping brain is not only managing thoughts and feelings, it is also coordinating the biological restoration that prepares the body for another day of activity. The quiet work of the night continues. While sleep reshapes memories and adjusts emotional responses, another quieter process unfolds at the same time. The body begins repairing itself. Muscles relax, cellular processes shift, hormones rise and fall in carefully timed patterns. The sleeping state creates biological conditions that allow tissues to restore, maintain, and recalibrate themselves after the demands of the day. What appears to be inactivity is, in reality, an elaborate maintenance cycle. One of the most important signals involved in this process is growth hormone. Despite its name, growth hormone does not only influence childhood development. In adults, it plays a major role in tissue repair, protein synthesis, and cellular regeneration. During deep, slow-wave sleep, the pituitary gland releases a significant pulse of growth hormone into the bloodstream. This release is remarkably synchronized with the deepest stages of sleep. As large, slow brain waves ripple across the cortex, growth hormone secretion increases dramatically. These pulses help stimulate the repair of muscle fibers, the rebuilding of cellular structures, and the synthesis of proteins necessary for maintaining healthy tissues. Athletes often notice the practical consequences of this relationship. Physical training during the day places stress on muscles and connective tissue. During the following night, slow-wave sleep helps coordinate the repair process that strengthens those tissues. The body quietly rebuilds itself. But tissue repair is only one aspect of sleep's restorative work. The immune system also becomes more active during sleep. Immune cells communicate with one another through chemical signals known as cytokines. Certain cytokines increase during sleep and appear to help coordinate immune responses. These signals assist the body in identifying and responding to potential threats, such as infections or damaged cells. Research has shown that individuals who consistently obtain adequate sleep often show stronger immune responses to vaccinations compared with those who are sleep-deprived. In one set of experiments, participants who slept normally after receiving a vaccine developed significantly higher antibody levels than participants who were kept awake. Sleep appeared to strengthen immune memory. This finding highlights an important principle. The immune system does not work in isolation. It interacts closely with the brain and with sleep-regulating systems. During sleep, the body creates conditions that allow immune processes to function efficiently, detecting problems and coordinating defensive responses. Even minor sleep disruption can influence this balance. Another critical aspect of sleep's biological repair cycle involves metabolism. Metabolic systems regulate how the body uses energy, manages nutrients, and maintains stable blood chemistry. When sleep patterns are disrupted, these metabolic processes can become less stable. Some of the most detailed investigations into this relationship come from the work of the endocrinologist Eve Van Cauter at the University of Chicago. Van Cauter and her colleagues conducted extensive research examining how sleep deprivation influences hormonal systems and metabolic regulation. Their findings revealed that insufficient sleep can significantly alter glucose metabolism, the process through which the body regulates blood sugar. Participants who experienced restricted sleep showed reduced insulin sensitivity. Insulin is the hormone responsible for helping cells absorb glucose from the bloodstream. When insulin sensitivity decreases, the body must produce more insulin to achieve the same effect. Over time, this imbalance can place stress on metabolic systems and increase the risk of metabolic disorders. Van Cauter's research demonstrated that even a few nights of restricted sleep could produce measurable changes in glucose regulation. The body behaved in ways that resembled early stages of metabolic dysfunction. Sleep, it turned out, was closely connected to the body's energy balance. Hormonal systems involved in appetite regulation also respond to sleep duration. Two hormones, leptin and ghrelin, help control feelings of hunger and fullness. Leptin signals satiety, while ghrelin stimulates appetite. When sleep becomes restricted, leptin levels tend to decrease while ghrelin levels increase. The result is a biological shift that encourages increased hunger. Many people have experienced this effect firsthand. After a poor night of sleep, cravings for high-calorie foods often become stronger. The brain seeks additional energy, possibly as a way to compensate for the fatigue associated with sleep loss. This subtle hormonal shift illustrates how deeply sleep is woven into the body's regulatory systems. Sleep also influences cardiovascular health. The heart and blood vessels follow circadian rhythms and respond to hormonal changes that occur during sleep. Blood pressure tends to decrease during deep sleep, allowing the cardiovascular system to rest and recover from the demands of waking activity. When sleep is chronically disrupted, this nightly reduction in blood pressure may not occur consistently. Long-term studies have linked chronic sleep deprivation with increased risk of hypertension, cardiovascular disease, and other circulatory disorders. The mechanisms involved are complex, but they likely include persistent activation of stress-related systems within the body. Sleep allows those systems to quiet. Without sufficient sleep, the body may remain in a prolonged state of physiological alertness. Over time, this constant activation places strain on organs and tissues that were designed to experience regular cycles of activity and rest. The biological repair cycle of sleep therefore extends far beyond the brain. Hormonal signals, immune activity, metabolic regulation, and cardiovascular function all interact within the sleeping body. Each night, multiple systems enter coordinated states that promote restoration. The brain orchestrates these processes through complex interactions between neural circuits and endocrine signals. Slow-wave sleep appears particularly important for physical restoration, while other stages contribute to emotional processing and memory consolidation. Different stages of sleep perform different tasks. Together, they form a biological maintenance program that unfolds night after night. Yet even this impressive list of restorative functions does not capture the full story. In recent years, scientists have discovered that sleep may also serve another crucial role within the brain itself. During sleep, the brain appears to activate a specialized system responsible for clearing away metabolic waste products that accumulate during waking activity. This discovery revealed that sleep may function not only as a time for repair and regulation, but also as a time for neurological cleaning. The brain, it turns out, takes out the trash while its owner sleeps. While the sleeping body repairs tissues, stabilizes metabolism, and reorganizes memories, something equally important unfolds deep within the brain itself. Throughout the day, the brain works continuously. Neurons fire billions of times, chemical reactions occur. Across countless synapses, and metabolic processes generate byproducts as energy is consumed. Like any active system, the brain produces waste. For a long time, neuroscientists wondered how the brain removed these waste products. Most organs in the body rely on the lymphatic system, a network of vessels that carry away cellular debris and excess fluid. But the brain appears to lack conventional lymphatic vessels within its tissue. This raised an important question. If the brain works so hard, how does it clean itself? A remarkable answer began to emerge in the early 21st century through research led by the neuroscientist Maiken Nedergaard at the University of Rochester Medical Center. Nedergaard and her colleagues discovered what is now known as the glymphatic system, a previously unrecognized mechanism that helps clear metabolic waste from the brain. The name glymphatic combines two ideas. The system involves glial cells, which are support cells surrounding neurons, and it functions in a way that resembles the lymphatic system elsewhere in the body. This discovery revealed that the brain possesses its own specialized cleaning network. The glymphatic system works by circulating cerebrospinal fluid through the brain's tissue. Cerebrospinal fluid is a clear liquid that surrounds the brain and spinal cord, acting as both a cushion and a transport medium. During waking life, this fluid moves around the brain in relatively modest amounts. During sleep, however, something unusual happens. The circulation of cerebrospinal fluid increases dramatically. In experiments conducted at the University of Rochester Medical Center, researchers used advanced imaging techniques to observe fluid movement within the brains of sleeping animals. They found that when the animals entered deep, slow-wave sleep, channels between brain cells appeared to expand. This expansion allowed cerebrospinal fluid to flow more freely through the tissue. As the fluid moved through these microscopic spaces, it carried away metabolic waste products that had accumulated during waking activity. Proteins, fragments of cellular debris, and other molecules were flushed from the brain and eventually transported into the bloodstream for removal. The sleeping brain was cleaning itself. The increase in fluid movement during sleep is striking. Measurements suggest that the efficiency of waste clearance can increase significantly when the brain enters deep sleep compared with when it is awake. In essence, sleep opens the cleaning channels. The brain becomes a flowing system. One of the most important substances removed by the glymphatic system is a protein known as beta amyloid. Beta amyloid forms naturally during normal brain activity, but when it accumulates excessively, it can form sticky plaques between neurons. These plaques are one of the characteristic features observed in the brains of individuals with Alzheimer's disease. The discovery that sleep helps clear beta amyloid has attracted considerable scientific interest. If the glymphatic system removes this protein during sleep, then chronic sleep disruption could potentially interfere with that cleaning process. Over many years, insufficient sleep might allow waste products to accumulate more easily within brain tissue. While research in this area is still ongoing, several studies have suggested connections between long-term sleep disruption and increased risk of neurodegenerative conditions. The relationship is complex and involves many factors, but the link highlights the importance of the brain's nightly maintenance cycle. sleep may help protect neural health by keeping the brain's internal environment clean. The mechanism behind this cleaning system involves more than just fluid flow. Glial cells play an important role in regulating the spaces through which cerebrospinal fluid travels. These cells surround blood vessels and form channels that guide the movement of fluid through the brain's tissue. When the brain enters deep, slow-wave sleep, the activity of neurons decreases slightly, and the spaces between cells appear to expand. This subtle expansion creates room for fluid to circulate. In contrast, during waking activity, the brain remains densely active, and the spaces between cells are smaller. The glymphatic system still operates during waking hours, but it appears to work far more efficiently during sleep. The brain waits until nightfall to perform its most thorough cleaning. Researchers continue to explore how this system interacts with other biological processes. Blood flow, neural activity, and cerebrospinal fluid dynamics all appear to coordinate with sleep rhythms. The slow oscillations of deep sleep may even help push fluid through the brain in gentle pulses. The sleeping brain behaves almost like a breathing organ. Fluid moves in and out, waste is carried away, and the environment surrounding neurons becomes refreshed. The discovery of the glymphatic system also reminds scientists that sleep is not simply a passive shutdown of consciousness. Instead, sleep creates a biological state that allows specialized maintenance processes to operate efficiently. The brain schedules its cleaning work for the night. This insight has broadened the scientific understanding of why sleep is so essential. Without regular sleep, the brain may struggle to maintain its internal environment. Metabolic byproducts could accumulate, cellular processes could become less stable, and neural systems might gradually lose efficiency. Sleep, therefore, serves as a period of neurological renewal. Every night the brain reorganizes memories, regulates emotional responses, repairs tissues, and clears away the chemical residue left behind by waking activity. Each process contributes to the stability and resilience of the nervous system. Yet the question remains, if sleep performs so many essential tasks, how did such a vulnerable state evolve in the first place? Becoming unconscious for hours at a time would seem risky for animals living in unpredictable environments, and yet nearly every complex animal on Earth sleeps. Understanding why evolution preserved this behavior leads to a deeper question about the biological purpose of sleep itself. Sleep performs many important tasks. It strengthens memories, regulates emotions, repairs tissues, and clears metabolic waste from the brain. Yet these discoveries lead to a deeper and more puzzling question. Why did sleep evolve in the first place? From a survival perspective, sleep appears almost reckless. An animal that becomes unconscious for hours at a time loses awareness of its surroundings. It cannot search for food, defend itself effectively, or respond quickly to danger. In environments filled with predators and competition, becoming vulnerable seems like a risky strategy, and yet sleep appears nearly universal across the animal kingdom. Birds sleep, mammals sleep, reptiles and fish show sleep-like states. Even certain insects and simple organisms display patterns resembling sleep behavior. Evolution rarely preserves traits that carry serious risks unless those traits provide powerful advantages. The fact that sleep has persisted across millions of years suggests that its benefits must be substantial enough to outweigh its dangers. Scientists have proposed several explanations for how this balance might work. One of the earliest evolutionary ideas is known as the adaptive inactivity theory. According to this hypothesis, sleep evolved as a way of conserving energy and avoiding unnecessary activity during periods when being awake would provide little advantage. Many environments contain predictable cycles of light and darkness. During nighttime, visibility declines and certain resources become harder to locate. For animals that rely heavily on daylight vision, moving around in darkness may be inefficient or dangerous. Under these conditions, remaining inactive for part of the daily cycle may actually improve survival. Sleep could therefore represent a form of strategic downtime. Rather than wandering aimlessly when conditions are poor, animals conserve energy and wait for more favorable circumstances. The body reduces metabolic demands, and activity resumes when environmental conditions improve. This interpretation treats sleep not as wasted time but as adaptive scheduling. Biological rhythms coordinate behavior with the structure of the environment. Yet energy conservation alone does not fully explain the complexity of sleep. Many animals that sleep still maintain substantial brain activity during sleep stages, and the brain continues performing numerous tasks throughout the night. Another perspective focuses on how sleep varies across species. The neuroscientist Jerome Siegel at the University of California, Los Angeles studied sleep patterns in many different animals. His research revealed that sleep duration differs dramatically across species, depending on ecological conditions. Some animals sleep only a few hours each day, while others sleep for most of the 24-hour cycle. Large grazing animals such as elephants and giraffes tend to sleep very little, sometimes only a few hours per day. Because these animals must spend long periods eating to sustain their body size, extended sleep may not be practical. Predators that face fewer threats often sleep longer. Lions, for example, may sleep for well over half of the day. Their hunting strategy involves bursts of intense activity followed by long periods of rest. Small mammals that face constant predation pressures often show fragmented sleep patterns, allowing them to wake quickly if danger approaches. These patterns suggest that sleep evolved in ways that adapt to each species' ecological niche. Sleep duration and structure appear shaped by environmental demands rather than following a single universal schedule. Yet another hypothesis places the focus on the brain itself. The neural plasticity theory proposes that sleep evolved primarily to support learning and brain development. Neural plasticity refers to the brain's ability to change its structure and strengthen or weaken connections between neurons based on experience. Young animals, whose brains are developing rapidly, tend to spend especially large amounts of time in sleep, particularly in rapid eye movement sleep. Human infants, for example, spend roughly half of their sleep time in rapid eye movement sleep. This stage is associated with intense brain activity and may play a role in forming and stabilizing neural connections. As the brain matures, the proportion of rapid eye movement sleep gradually declines. This pattern suggests that sleep may help shape neural circuits during development. The brain uses sleep to reorganize connections formed during waking experiences, strengthening useful patterns while trimming away weaker ones. Over time, this process helps refine the networks that support perception, memory, and behavior. Sleep becomes a tool for sculpting the brain. More recent work has explored how energy flows through the brain during sleep and wakefulness. One proposal, developed by the neuroscientist Władysław Wierzchowski, examines how sleep may redistribute metabolic resources across neural circuits. During waking activity, neurons consume large amounts of energy as they process information and transmit signals. Some neural networks may become particularly active depending on the task performed during the day. According to the energy allocation model, sleep allows the brain to rebalance these energy demands. Neural circuits that were heavily used during waking hours may receive metabolic support and repair during sleep, while less active circuits recover from periods of reduced activity. The sleeping brain redistributes resources to maintain long-term stability across its complex network. In this way, sleep may help ensure that the brain remains capable of efficient operation across successive days. Different theories highlight different aspects of sleep's purpose. Some emphasize energy conservation and environmental adaptation. Others focus on learning, neural plasticity, and metabolic maintenance. These explanations are not necessarily mutually exclusive. Sleep may have evolved because it performs many functions simultaneously. Evolution often favors solutions that solve several problems at once. A behavior that conserves energy, supports learning, maintains neural health, and regulates physiology would provide enormous survival advantages over time. The persistence of sleep across so many species suggests that its benefits are deeply embedded in biological systems, and yet the precise evolutionary origins of sleep remain partially mysterious. Because sleep leaves animals temporarily vulnerable, natural selection would only preserve it if the underlying advantages were powerful and unavoidable. The evidence increasingly suggests that the brain and body require sleep in order to function properly. Sleep is not merely optional rest. It is a fundamental biological process woven into the architecture of life itself. Looking across the animal kingdom reveals just how widespread and varied this process can be. Different species have evolved different strategies for achieving sleep, sometimes in ways that seem almost impossible from a human perspective. Some animals sleep with only half of their brain at a time. Others drift in and out of brief sleep episodes throughout the day. The diversity of these patterns provides new clues about how sleep works and why evolution shaped it the way it did. Sleep appears nearly universal among complex animals, but the ways different species experience sleep can be surprisingly diverse. Humans typically imagine sleep as a complete shutdown. The lights go out, consciousness fades, and the entire brain enters a coordinated pattern of rest. But nature has developed far more flexible approaches to the same biological requirement. Some animals sleep with half of their brain at a time. This remarkable phenomenon is known as unihemispheric slow-wave sleep. In this state, one hemisphere of the brain enters deep slow-wave sleep, while the other hemisphere remains awake and alert. The sleeping half of the brain displays the large, slow oscillations characteristic of deep sleep, while the waking half maintains activity patterns similar to those seen during alert behavior. The two halves of the brain take turns. Dolphins are among the most famous examples of this unusual strategy. As marine mammals, dolphins must regularly swim to the surface in order to breathe. If they were to fall fully unconscious for long periods, they would risk drowning. Unihemispheric sleep provides a solution. While one half of the dolphin's brain sleeps, the other half remains sufficiently alert to control breathing and maintain basic swimming movements. After a period of time, the roles reverse, allowing the previously awake hemisphere to rest while the other resumes monitoring the environment. Birds have evolved similar strategies. Certain species of migratory birds can sleep while flying, allowing them to continue long-distance journeys without fully stopping to rest. Research using electroencephalography recordings has shown that birds may briefly engage one hemisphere in slow-wave sleep, while the other hemisphere remains attentive to flight control. Even birds resting on land sometimes keep one eye open, because each eye primarily communicates with the opposite hemisphere of the brain. A bird can sleep with one half of its brain, while the other half watches for predators. Nature rarely wastes opportunities for clever design. While marine mammals and birds demonstrate dramatic examples of sleep flexibility, simpler organisms also show patterns that resemble sleep. These observations have broadened scientific understanding of how deeply rooted sleep behavior may be in evolutionary history. One particularly influential line of research comes from experiments involving fruit flies. The fruit fly, known scientifically as Drosophila melanogaster, might not seem like an obvious subject for sleep research. Yet these tiny insects have proven remarkably useful for understanding biological rhythms and neural behavior. In the early 2000s, the neuroscientist Paul Shore and his colleagues conducted experiments examining rest patterns in fruit flies. They observed that fruit flies regularly enter periods of inactivity, during which their responsiveness to external stimuli decreases. During these periods, the flies become harder to awaken, and their brain activity changes in ways that resemble sleep-like states. Even more intriguingly, when fruit flies are deprived of these rest periods, they later compensate by increasing the duration of their inactivity. This rebound effect closely mirrors the sleep rebound seen in mammals after sleep deprivation. The tiny insect brain appears to follow rules similar to those governing human sleep. Genetic experiments with fruit flies have also revealed specific genes that influence sleep duration and regulation. Because fruit flies are relatively easy to study genetically, researchers have been able to identify molecular pathways that help control sleep behavior. These findings suggest that some components of sleep regulation may be shared across many branches of the animal kingdom. Even more surprising discoveries have emerged from organisms that lack complex brains altogether. Researchers studying jellyfish have observed sleep-like states in certain species, including the upside-down jellyfish known as Cassiopeia. These animals possess a decentralized nerve net rather than a centralized brain, yet they still display patterns of reduced activity that resemble sleep. When jellyfish enter these inactive periods, their pulsation rate slows and their responsiveness to stimuli decreases. If these periods are repeatedly interrupted, the jellyfish later show longer periods of inactivity, suggesting a rebound effect similar to sleep deprivation in more complex animals. The existence of sleep-like states in such simple organisms raises fascinating questions about the origins of sleep. Sleep may not have evolved solely within animals possessing large brains. Instead, the behavior may represent a much older biological strategy related to cellular maintenance, energy management, or neural coordination. Sleep may be deeply embedded in the foundations of animal life. Across the animal kingdom, the amount of time spent sleeping varies dramatically. Large herbivores such as elephants often sleep relatively little. Observations of African elephants suggest that they may sleep only a few hours per day, often while standing. But as these animals must spend long periods feeding and traveling to locate vegetation, extended sleep would limit their ability to obtain sufficient energy. At the opposite extreme are animals such as bats. Certain bat species may sleep for well over half of each day, sometimes approaching 20 hours of sleep within a 24-hour cycle. Hanging safely within sheltered roosts, bats face fewer immediate threats from predators and can afford to remain inactive for long periods. These differences highlight an important principle. Sleep duration is shaped by ecological demands. Animals balance the benefits of sleep against the risks and opportunities present in their environment. Predators, prey, feeding patterns, body size and habitat all influence how sleep evolves within a species. Despite these differences, the core biological need for sleep appears remarkably consistent. When animals are deprived of sleep, many species exhibit clear rebound effects. They seek opportunities to recover lost sleep, suggesting that the underlying physiological processes remain essential. Sleep cannot simply be skipped across Species as different as mammals, birds, insects, and jellyfish. Periods of reduced activity and altered neural function appear repeatedly. The details vary, but the general pattern persists. Life, it seems, requires cycles of activity and restoration. Studying how animals sleep has therefore provided valuable clues about the deep evolutionary roots of this behavior. By examining sleep in creatures ranging from fruit flies to dolphins, scientists gain insight into the fundamental processes that sleep supports. These discoveries also remind us that sleep is not always fragile. In some species it is remarkably adaptable, but in humans, sleep can sometimes become disrupted in ways that interfere with its many biological roles. When the delicate balance of sleep regulation breaks down, the consequences can affect both mental and physical health. Understanding these disruptions brings attention to the modern study of sleep disorders and the challenges they create for the sleeping brain. Sleep is often described as natural and effortless. For most people, it arrives quietly each night, guided by internal clocks and chemical signals that have evolved over millions of years. Yet the systems responsible for producing sleep are delicate. They rely on complex coordination between brain circuits, hormones, breathing patterns, and environmental cues. When those systems fail, sleep can become fragmented, unpredictable, or even impossible. Modern sleep medicine has revealed that disturbances in sleep are not simply inconveniences. They often reflect disruptions within the neurological mechanisms that normally regulate sleep and wakefulness. Understanding these conditions has helped scientists appreciate just how intricate the biology of sleep truly is. One striking example is narcolepsy. Narcolepsy is a neurological disorder in which the brain loses its ability to maintain stable boundaries between wakefulness and rapid eye movement sleep. Individuals with narcolepsy may experience sudden episodes of overwhelming sleepiness during the day, even after apparently sufficient nighttime rest. In some cases, rapid eye movement sleep intrudes directly into waking life. This can lead to vivid dreamlike hallucinations, sudden muscle weakness known as cataplexy, or temporary paralysis when falling asleep or waking. These experiences may feel strange or unsettling, but they provide important clues about how sleep normally works. Research conducted at the Stanford Sleep Center helped uncover the biological basis of narcolepsy. Scientists, including Emmanuel Mignot, discovered that many cases of narcolepsy involve the loss of neurons that produce a neurochemical known as hypocretin, sometimes called orexin. Hypocretin plays a critical role in stabilizing wakefulness. Neurons that release hypocretin form networks that communicate with multiple brain regions responsible for maintaining alertness. When these neurons function normally, they help keep the brain in a stable waking state. In individuals with narcolepsy, many of these neurons are missing or inactive. Without hypocretin, the brain struggles to maintain clear boundaries between sleep stages and wakefulness. The result is a system in which sleep and dreaming processes intrude unpredictably into daytime consciousness. Narcolepsy therefore reveals something important about normal sleep regulation. Sleep and wakefulness are not simply opposites. They are states carefully stabilized by networks of neurons that must continuously balance competing signals. Another common sleep disorder arises from a completely different biological mechanism. Sleep apnea involves repeated interruptions of breathing during sleep. In obstructive sleep apnea, the muscles that normally keep the airway open relax too much while the person is asleep. As a result, the airway partially or completely collapses, temporarily blocking airflow. The sleeping brain senses this disruption and briefly awakens the body in order to restore breathing. These awakenings are often so brief that the person does not remember them. Yet they may occur dozens or even hundreds of times throughout the night. Each interruption fragments the architecture of sleep, preventing the brain from maintaining continuous cycles of restorative sleep stages. The consequences can extend far beyond simple fatigue. Because oxygen levels repeatedly fall and rise during episodes of sleep apnea, the cardiovascular system experiences significant stress. Over time, untreated sleep apnea has been linked to increased risk of hypertension, heart disease, and metabolic disturbances. Sleep, which normally protects the body, becomes disrupted in ways that strain multiple physiological systems. Another widespread sleep disorder involves the difficulty of falling asleep or staying asleep in the first place. Chronic insomnia affects millions of people worldwide and illustrates how psychological and neurological processes can interact with sleep regulation. Individuals experiencing chronic insomnia often report lying awake despite feeling physically tired. Brain imaging studies suggest that certain wake-promoting circuits may remain unusually active in these individuals, preventing the brain from fully transitioning into sleep. In some cases, heightened activity in stress-related systems may keep the brain in a state of alertness, even when environmental conditions are quiet and safe. The mind becomes stuck between states. Part of the brain seeks sleep. Another part refuses to let go of wakefulness. Researchers studying insomnia have also observed changes in circadian rhythm signals and sleep pressure regulation. The delicate balance between these systems can become misaligned, making it harder for the body to coordinate the timing of sleep. Sleep medicine has developed a variety of approaches to address these challenges. Organizations such as the American Academy of Sleep Medicine have established diagnostic standards and treatment guidelines that help clinicians identify specific sleep disorders. Advances in monitoring technology now allow physicians to observe breathing patterns, brain activity, heart rhythms, and muscle movements throughout the night. These detailed measurements reveal how sleep systems behave in real time. Treatment approaches vary depending on the underlying cause. For sleep apnea, one of the most widely used therapies involves continuous positive airway pressure devices. These devices deliver gentle air pressure through a mask, helping keep the airway open during sleep. For narcolepsy, medications that influence wake-promoting neurotransmitters can help stabilize sleep and wakefulness patterns. Chronic insomnia is often treated using behavioral therapies that help retrain the brain's sleep regulation systems. One widely used method is cognitive behavioral therapy for insomnia, which focuses on adjusting sleep habits, environmental cues, and mental associations related to bedtime. The goal is to restore the brain's natural ability to transition smoothly into sleep. Emerging technologies are also being explored. Some researchers are investigating forms of neural stimulation that influence specific brain circuits involved in sleep regulation. Others are developing wearable devices capable of monitoring sleep patterns continuously, allowing individuals to better understand their own sleep rhythms. These advances highlight how rapidly the field of sleep science is evolving. Yet even as medicine improves its ability to treat sleep disorders, scientists continue to explore another important aspect of sleep that often surprises people. Human sleep patterns are not identical for everyone. Even when sleep systems function normally, individuals may experience significant variation in when and how they sleep. Some people naturally wake before sunrise, while others feel most alert late at night. These differences reflect deeper variations in the timing systems that regulate sleep itself. Although sleep follows universal biological principles, the way humans experience sleep varies remarkably from one person to another. Some people wake naturally before sunrise, feeling alert and ready to begin the day while the sky is still dim. Others feel most energetic long after sunset, when the world grows quiet and many people are already asleep. These differences are so familiar that they often acquire simple labels: morning people, night owls. But behind these everyday descriptions lies a deeper biological concept known as chronotype. Chronotype refers to an individual's natural tendency toward particular sleep and wake times within the 24-hour circadian cycle. Rather than being determined solely by habit or discipline, chronotypes are strongly influenced by genetics and internal biological clocks. The brain's circadian timing system is built from networks of genes that regulate rhythmic activity inside cells. These genes generate biochemical cycles that repeat roughly every 24 hours, helping coordinate sleep, hormone release, body temperature, and metabolism. Among the most important components of this system are the clock genes. Research into these genes has revealed that small variations in their structure can influence when a person naturally feels sleepy or alert. One influential line of investigation examined families in which several members consistently fell asleep and woke unusually early compared with the general population. Scientists studying these families discovered mutations affecting a gene known as period 2, often called PER2. This gene plays a central role in regulating the timing of circadian rhythms. When the gene functions slightly differently, the internal clock can run faster than usual. The result is a biological preference for earlier sleep and wake times. Other variations in clock genes appear to push rhythms in the opposite direction, creating individuals whose internal clocks run slightly longer than the 24-hour day. These individuals may feel naturally inclined to stay awake later at night and wake later in the morning. Neither pattern is inherently better than the other. They simply represent different expressions of the same biological timing system. Chronotype also changes across the human lifespan. Sleep patterns that feel natural during childhood may shift dramatically during adolescence, only to change again later in adulthood. Infants provide one of the clearest examples of how sleep evolves with age. Newborn babies spend much of their time asleep, often sleeping well over half of each day. Their sleep occurs in short cycles scattered across the 24-hour period because their circadian rhythms are not yet fully developed. Rapid eye movement sleep occupies a large proportion of this early sleep. The developing brain appears to rely heavily on this stage as neural circuits grow and refine their connections. As children grow older, sleep gradually becomes more consolidated at night. During adolescence, however, the internal clock often shifts later. Teenagers frequently experience a biological delay in their circadian rhythm, causing them to feel naturally alert later at night and sleepy later in the morning. This shift can sometimes conflict with early school schedules, creating a mismatch between biological timing and social expectations. By adulthood, many people settle into relatively stable sleep patterns, though individual chronotypes continue to vary widely. Later in life, sleep often becomes lighter and more fragmented. Older adults may experience earlier bedtimes and earlier wake times, along with gradual reductions in deep, slow-wave sleep. These changes reflect alterations in circadian signals, hormonal rhythms, and neural activity within sleep-regulating systems. Biology is not the only influence shaping human sleep patterns. Culture and environment also play powerful roles. Before the widespread use of artificial lighting, human activity followed natural cycles of daylight and darkness more closely. Evening darkness signaled the body to begin producing melatonin, a hormone that helps promote sleep. In many pre-industrial societies, sleep sometimes occurred in segmented patterns. Historical records suggest that people occasionally experienced a period of first sleep shortly after nightfall, followed by a quiet waking interval, and then a second sleep later in the night. During the waking interval, Individuals might read, pray, talk quietly, or simply rest before returning to sleep. The modern concept of a single uninterrupted night of sleep may therefore be partly shaped by cultural expectations rather than strict biological necessity. The arrival of artificial lighting transformed this relationship between humans and the night. Electric light extended waking hours far beyond sunset, allowing work, entertainment, and social activity to continue late into the evening. While this innovation brought enormous benefits, it also altered the signals that guide circadian rhythms. Light is the most powerful environmental cue controlling the brain's internal clock. Specialized cells in the retina detect light and transmit signals to the suprachiasmatic nucleus within the hypothalamus, the brain's master circadian clock. Exposure to bright light in the evening can delay the release of melatonin and shift the timing of the internal clock later. In modern environments filled with screens, lamps, and illuminated cities, this effect can become quite pronounced. Electronic devices such as smartphones, tablets, and computers often emit light rich in blue wavelengths. These wavelengths are particularly effective at influencing circadian rhythms. When the eyes detect blue light in the evening, the brain may interpret it as a signal that daytime is continuing. Melatonin release is delayed. Sleepiness arrives later. Over time, frequent exposure to artificial light late in the evening can shift circadian timing further away from the natural cycle of daylight and darkness. This misalignment between internal clocks and external schedules is sometimes referred to as social jet lag. The body feels as though it is living in a different time zone from the one imposed by daily obligations. Understanding these influences has become an important area of research in modern sleep science. Scientists are exploring how light exposure, work schedules, travel, and technology interact with the biological clock. Simple adjustments to lighting environments can sometimes help restore healthier sleep timing. Exposure to bright natural light during the morning and reduced artificial light in the evening can help reinforce the body's natural rhythms. The internal clock responds continuously to the signals it receives from the environment. Human sleep therefore reflects a dynamic interaction between biology, development, and culture. Genetic variation shapes chronotype. Age influences sleep architecture. Environmental light cues adjust the timing of circadian rhythms. Together, these forces create the diverse patterns of sleep observed across human populations. Even with this growing understanding, however, sleep science continues to uncover new questions. The brain's nightly processes remain complex, and researchers are still exploring how sleep influences cognition, health, and long-term brain function. The story of sleep is far from complete. New discoveries continue to reveal just how intricate the sleeping brain truly is. Sleep science has progressed enormously over the past century, yet many of its most intriguing discoveries have emerged only in recent decades. The deeper researchers look into the sleeping brain, the more complex the picture becomes. For a long time, sleep was thought to be a global state. The assumption was simple: either the brain was awake or the brain was asleep. Entire networks of neurons were believed to switch between these conditions together, much like a city turning its lights on or off. Modern neuroscience has revealed that the reality is far more nuanced. One emerging idea is known as local sleep. According to this concept, small regions of the brain can briefly enter sleep-like states, even while the rest of the brain remains awake. In other words, sleep may not always occur all at once. Different parts of the brain can rest at different times. Evidence for local sleep began appearing in experiments examining neural activity during periods of prolonged wakefulness. researchers noticed that individual groups of neurons sometimes displayed patterns of activity that resembled sleep, even when the animal or person was still awake and behaving normally. These brief episodes were subtle, but measurable. One of the scientists who helped explore this phenomenon is Władysław Wierzchowski, whose work has examined how cortical neurons behave during extended wakefulness. By recording the electrical activity of neurons in the cerebral cortex, Wierzchowski and colleagues discovered that some neurons occasionally fall silent for short periods, producing patterns similar to those seen during deep slow-wave sleep. These brief pauses can occur even while the individual remains awake. From the outside, the person may appear alert, yet at the microscopic level, small networks of neurons are momentarily entering sleep-like states. This discovery may help explain familiar experiences, moments when attention briefly slips, times when a person reads the same sentence several times without absorbing its meaning, episodes when concentration fades despite conscious effort. The brain may be experiencing tiny fragments of sleep. Local sleep suggests that sleep pressure can build unevenly across different brain regions, depending on how heavily those regions were used during waking activity. Areas that worked hardest during the day may be more likely to enter brief restorative pauses. The brain distributes rest where it is most needed. Another frontier in sleep research explores how the sleeping brain might refine its internal models of the world. Some theoretical neuroscientists propose that sleep allows the brain to reorganize predictive frameworks that guide perception and decision-making. These ideas connect sleep with the broader concept of predictive processing. According to predictive processing models, the brain continuously generates predictions about the environment and updates those predictions based on incoming sensory information. During waking life, sensory input from the outside world constrains these predictions. During sleep, however, sensory input becomes minimal. Without constant external signals, the brain may explore its own internal models more freely. Memories, experiences, and associations can be reorganized without the immediate pressure of responding to the external world. Sleep may therefore provide a unique computational environment. Within this environment, the brain can refine its predictions about reality, strengthen useful patterns of interpretation, and discard less helpful ones. The dreamlike imagery sometimes experienced during sleep may represent fragments of this internal modeling process. The sleeping mind is not simply replaying the past, it may also be rehearsing possibilities. As sleep research expands, new technologies are helping scientists examine these processes in greater detail. One rapidly growing area involves the use of artificial intelligence to analyze sleep data. Sleep laboratories generate enormous amounts of information. Brain recordings, heart rhythms, breathing patterns, eye movements, and muscle activity are all monitored during sleep studies. Traditionally trained specialists examine these recordings manually in order to identify sleep stages and detect abnormalities. Artificial intelligence systems are now being trained to recognize patterns within these complex datasets. Machine learning algorithms can examine thousands of hours of sleep recordings and identify subtle patterns that might be difficult for human observers to detect. These systems can classify sleep stages, identify unusual neural activity, and help researchers discover relationships between sleep patterns and health outcomes. Artificial intelligence does not replace human expertise, but it provides powerful tools for exploring large-scale data. With these tools, scientists can study sleep across thousands of individuals rather than just small laboratory groups. Large research initiatives are making this possible. Organizations such as the National Institutes of Health have funded extensive projects designed to investigate sleep patterns across entire populations. These studies combine wearable monitoring devices, brain imaging technologies, and genetic analysis to understand how sleep interacts with health, behavior, and disease risk. Participants may wear devices that track movement, heart rate, and light exposure over long periods. These measurements provide detailed information about real-world sleep patterns outside the controlled environment of a laboratory. When combined with medical data and cognitive testing, researchers can explore how sleep influences everything from memory performance to cardiovascular health. The scale of these projects reflects the growing recognition that sleep is central to nearly every aspect of human biology. And yet, despite all of these advances, some of the most basic questions about sleep remain unanswered. Why does the brain require sleep at all? Why do dreams occur? Why do different species sleep in such dramatically different ways? Each new discovery reveals another layer of complexity. Sleep appears to regulate memory, emotion, metabolism, immune function, neural maintenance, and cognitive performance. It shapes development and supports learning. It protects long-term brain health and influences daily behavior. Still, scientists continue to uncover new mechanisms that were previously hidden. The sleeping brain remains one of biology's most fascinating frontiers, and as research continues to advance, the mysteries surrounding sleep grow both clearer and deeper at the same time. After centuries of observation and decades of modern neuroscience, sleep remains one of the most familiar experiences in human life, and yet it is still not fully understood. Researchers now know far more about sleep than at any point in history. The architecture of sleep stages has been mapped in detail. The chemical signals that regulate sleep have been identified. Scientists have observed how sleep influences memory, emotion, metabolism, and neural maintenance. But beneath these discoveries lies a deeper mystery. Why must sleep exist at all? One of the most puzzling aspects of sleep is its near universality across the animal kingdom. Mammals sleep. Birds sleep. Reptiles display sleep-like states. Fish rest in ways that resemble sleep. Even simple organisms such as jellyfish show periods of reduced responsiveness that follow patterns similar to sleep behavior. Life appears to require cycles of rest and activity, and yet the amount of sleep required varies enormously. Elephants may sleep only a few hours each day. Bats can sleep for most of the 24-hour cycle. Some migratory birds sleep in brief episodes while flying. Marine mammals allow only half of the brain to sleep at a time. If sleep performs a single universal function, such dramatic variation becomes difficult to explain. This raises an intriguing possibility. Sleep may not serve just one purpose. Instead, it may perform several overlapping roles at once. Modern research has revealed a long list of processes that occur during sleep. Memory consolidation strengthens learning by replaying neural activity patterns. Emotional processing adjusts the intensity of experiences stored in memory. Growth hormone supports tissue repair, immune signaling, coordinates defensive responses. The glymphatic system removes metabolic waste from brain tissue. Each of these functions appears important, yet none of them alone fully explains why sleep is absolutely necessary for survival. Experiments involving prolonged sleep deprivation demonstrate how essential sleep truly is. Animals deprived of sleep eventually experience severe physiological disturbances, including immune dysfunction, metabolic instability, and cognitive impairment. Eventually, the body simply cannot maintain normal function. Sleep is not optional, but precisely which processes make sleep indispensable remains an open question. Some scientists believe that sleep evolved primarily to support neural plasticity. According to this perspective, the brain requires regular periods of reduced sensory input in order to reorganize neural connections formed during waking experiences. Without sleep, neural circuits may become overloaded or inefficient. Other researchers emphasize metabolic restoration. The brain consumes a large portion of the body's energy, and its activity generates metabolic byproducts that must be removed. Sleep may provide the conditions necessary for the glymphatic system and other maintenance processes to operate efficiently. Still others highlight memory consolidation as the central function. The hippocampus replays recent experiences, transferring information into long-term cortical storage networks. Without this nightly reorganization, learning and memory formation may become less stable. Each theory captures part of the picture, but none seems to explain everything. The truth may lie in the interaction between multiple biological systems. Sleep may serve as a coordinated maintenance period in which the brain and body carry out several forms of repair and reorganization simultaneously. Evolution may have shaped sleep as a multi-purpose solution. Understanding this complexity is challenging because the sleeping brain remains difficult to observe directly. Although modern technologies such as electroencephalography, functional magnetic resonance imaging, and neural recording techniques provide valuable information, each method has limitations. Electroencephalography measures electrical activity at the surface of the scalp, offering broad patterns of brain rhythms but limited spatial detail. Functional magnetic resonance imaging reveals changes in blood flow associated with neural activity, yet its temporal resolution is slower than the rapid electrical events occurring within neurons. Direct recordings from individual neurons provide detailed information, but can only be performed in limited experimental settings. The brain contains billions of neurons interacting simultaneously across many networks. Capturing this complexity during sleep remains a formidable scientific challenge. Genetics provides another promising avenue for understanding sleep. Researchers studying families with unusual sleep patterns have identified mutations in genes that influence sleep duration and timing. For example, mutations affecting the DEC2 gene have been associated with individuals who naturally sleep fewer hours than average, without apparent negative health effects. These rare individuals appear to function normally despite sleeping significantly less than most people. Studying such genetic variations may help scientists identify the molecular pathways that regulate sleep need. But even here, the story is complex. Sleep duration varies widely across individuals, and environmental factors such as light exposure, stress, and daily activity also influence sleep patterns. Genes provide part of the explanation, but they do not tell the entire story. The deeper scientists look, the clearer it becomes that sleep is not a single process, but a constellation of interacting systems. Neural circuits, hormones, metabolic pathways, and environmental signals all converge to shape the sleeping state. The night becomes a period when these systems operate together in coordinated rhythms. Memory is reorganized, emotions are recalibrated, tissues are repaired, waste products are removed. And yet even this long list of processes may not capture the full significance of sleep. Some functions may still remain undiscovered. As research continues, scientists may uncover new roles for sleep that have not yet been imagined. Each new experiment reveals another piece of the puzzle, while also reminding researchers how much remains unknown. Sleep, despite being one of the most familiar parts of daily life, still carries an element of mystery. Every night, the brain enters a state that is both essential and only partially understood. And that quiet nightly journey leads naturally to a final reflection on what the sleeping brain is truly doing while consciousness fades. Every night, something remarkable happens. Without conscious effort, without deliberate control, the brain begins guiding the body into a completely different biological state. Awareness fades, muscles relax, the constant flow of sensory information from the outside world gradually quiets. Sleep begins. For most people, this transition feels simple and ordinary. It is a familiar rhythm repeated every evening, often taken for granted because it happens so reliably. Yet beneath that ordinary experience lies one of the most intricate biological processes in the human body. Sleep is not the absence of activity. It is a carefully orchestrated transformation. Across the brain, networks of neurons shift their patterns of communication. Chemical messengers change their balance. Circadian signals align with accumulated sleep pressure. Brain rhythms slow, synchronize, and then evolve again as the night progresses through its repeating cycles. Each stage of sleep performs different tasks. Slow-wave sleep brings deep, synchronized oscillations across the cortex. During these periods, the brain quiets its firing patterns while strengthening newly formed memories and releasing hormones that support tissue repair. The body restores itself while the mind organizes information gathered throughout the day. Later in the night, rapid eye movement sleep emerges. The brain becomes active again in new ways. Visual regions ignite with activity. Emotional centers process experiences. Dream imagery unfolds while the body remains safely still, protected by neural circuits that temporarily inhibit movement. The sleeping mind revisits memories, reshapes emotions, and explores associations that might never arise during waking thought. At the same time, quieter systems are working behind the scenes. Cerebrospinal fluid circulates through the brain's glymphatic pathways, carrying away metabolic waste that accumulates during waking life. Immune signals strengthen the body's defenses. Hormonal systems rebalance metabolism and energy use. Throughout the night, countless processes unfold simultaneously. What appears to be rest is actually a period of extraordinary coordination. Evolution has refined this system over millions of years. Across the animal kingdom, sleep appears in many forms, from the unihemispheric sleep of dolphins to the brief rest cycles of insects. The details vary, but the pattern remains consistent. Life requires periods of restoration. The human brain, perhaps the most complex structure known in biology, depends especially on this nightly maintenance. Without sleep, cognitive performance declines. Emotional balance becomes unstable. Physical health begins to suffer. Sleep sustains the delicate equilibrium that allows the brain and body to function day after day. And yet, even with all that science has revealed, sleep still holds mysteries. Researchers continue exploring why sleep varies between species, how genes influence chronotypes, and how the brain reorganizes its internal networks during the night. New technologies are beginning to uncover details that were invisible only a generation ago. Each discovery deepens the understanding of how profoundly sleep shapes human life. Still, the most remarkable aspect of sleep may be how quietly it happens. The nightly transition into sleep requires no conscious planning. No deliberate commands are issued. The brain simply follows rhythms that have evolved over immense spans of time, guiding the body through a sequence of states that restore and maintain its systems. Night after night, this quiet machinery performs its work. Memories are strengthened, emotions are softened, cells are repaired, waste is cleared away. By morning, the brain emerges renewed, ready once again to interpret the world, learn new information, and navigate the countless experiences that fill each day. Sleep rarely announces its importance, but without it, the entire structure of waking life would slowly begin to unravel. So when the mind finally drifts towards sleep tonight, it will not simply be switching off. It will be entering one of biology's most remarkable processes, a silent collaboration of brain rhythms, chemical signals, and evolutionary design that continues working long after consciousness fades.